Greg Fisher, I'm the Facilities Operations Manager up at NADF. Been there since the beginning, and previously was a fisheries research biologist for the tribe here, so I've been here for almost 30 years and enjoy it quite a bit. The only thing I don't enjoy is not being able to fish today. So it's really nice out, and the cohos and the browns and the rainbows are popping, so every marina is really busy and all the boat slips are busy and everybody's out fishing, so it's crazy. You guys should have brought your boats. We could have done on the lake assessment, but. So on the way to work today, I stopped and talked to these guys. They had a successful morning hunting. Our squirrels are pretty big in northern Wisconsin. And I don't know where I got this, but it always gets me going. A couple years ago, I gave a session, a walleye culture session up in Ontario. It was a 3-day workshop with Alan Johnson and Bob Sommerfelt, and unfortunately Bob Sommerfelt dropped out. So Alan and I had to talk for 2-3 days straight. And about halfway through the session, I noticed all these people were kind of falling asleep and getting pretty bored, and so I just put up a bunch of hunting and fishing pictures and got them entertained for a little while. So today we're going to talk about cool water species, and it's timely because at the facility you guys are going to see our walleye and saugeye eggs hatching. We've got about 1.5 million eggs hatching right now. They started going full force yesterday. So we're scrambling a little bit because they hatch quickly and you have to keep things clean, and they also have to be stocked out into their tanks or into the ponds fairly quickly within a few days, and you're going to hear all about that and see it in action. We might even put you to work a little bit. But so I'm going to cover broodstock, spawning, egg and milk collection, fertilization, incubation, hatchery collection, or excuse me, hatching and collection of fry, enumeration of fry, stocking into tanks, and Emma's going to cover larval rearing. And we're going to do all that in like 30 minutes. Yeah. But we'll see what we can do. This is an overview of everything. So cool water broodstock, you know, we're talking about fish that the optimum water temperatures are probably between 50 and 76 degrees Fahrenheit. That's kind of just a basic range. I mean, of course, they go colder and they can go warmer, but optimum, the temperatures are in there. So we're not talking right now about cold water species or warm water species. Of course, cold water species would be much colder and warm water would be much warmer. So fairly short egg incubation, about 10 to 21 days, and that's based on water temperature. So we can speed them up and we can slow them down. We can kind of do what we need to in the hatchery, but we have found that if we speed them up too much, if we incubate them in less than 7 days, we see a lot of deformities and poor hatch out. So we kind of limit our incubation at the NADF facility to 10 days. We try not to go below that. And we have incubated them up to 21 days and even a little more. We've cold banked eggs before and with some success. So the species we're talking about are walleye, sauger, yellow perch, northern pike, muskie, white sucker, striped bass, things like that. For my talk, I'm concentrating mainly on walleye and sauger, but mainly walleye. Do you guys know what a sauger is? Everybody know what a sauger is? So it's a kissing cousin of a walleye. Literally, they are kissing cousins, and they will breed together, and we make saugeyes out of that. So we just we have saugeyes at the facility. A couple weeks ago, we had to go down to the Mississippi River and actually collect saugeyes, or excuse me, saugers with hook and line. It was a very difficult job. And I'm lucky enough to get to do that, and I took Jared, one of our techs that you'll meet later on. And Jared's the best fisherman ever. He lives, breathes, and eats fish, but he's allergic to fish. So it's really crazy. He doesn't eat fish, yeah. So he's the best person to have at the hatchery because he's never going to steal the fish. And I outfished Jared this last trip, and I don't let him forget it. I'll probably tell him today when you guys are there. So he's always out fishing me. He just successfully killed a 22-pound turkey yesterday, he told me. So yeah. So one thing about broodstock is your broodstock wild or is it captive? So of course wild is going out and collecting it, and captive would be that you have it in-house. And so if you have a captive broodstock, there's some things you can do or things you have to do to get them to spawn. So walleyes have to go through a winter period. So somewhere in that year, they have to go through a winter period where the temperature drops on them, and maybe there's some photoperiod and temperature manipulation. Do you guys know what photoperiod manipulation is? So fish are driven by the sunlight, right? The light period. So if we manipulate that light period, we can trick them into different seasons. So if we think about it, if you had fish in a dark tank in a dark room and you controlled the light and you control the temperature, they really don't know that it's not spring or summer. You can do whatever you want and trick them. So we've done that before. We've successfully spawned walleyes out of season just with photoperiod and temperature manipulation. We've been able to get them to spawn as early as like the end of January and as late as June. And we know that we can get them to spawn other times of the year because they're doing it in Europe with the European pike perch. If you guys have ever heard of that, it's also called zander. And a lot of times when you go and you have a walleye fry and it's like $6.99, you're probably eating zander. Yeah, or if you get walleye that doesn't taste good, it's zander. So the other thing you can do with both captive and wild broodstock is you can inject them with hormone and you can advance the spawning. So you can trick them with a hormone. And so we've used HCG, this human gonadotropin, whatever, HCG hormone, and 2 injections, 3 to 5 days apart at different rates. For females, we use 500 IUs per kilogram, and males 350 IUs per kilogram. We found that it's important to do 2 injections. 1 injection kind of primes them, and the 2nd injection finishes them. And we've had the most success with that, and we have successfully spawned fish that we like brought right in from the wild, injected them, and spawned them a few days later. But the eggs have to kind of have already went through that winter period. The fish have to be in that stage where they're getting ready to spawn. So walleyes are really ready to spawn by February and March, but they just don't spawn till April for us. But if we inject them, we can get them to spawn. So those are a couple things that you can do.
Where do you inject?
Where do we inject? If you can see that picture, right, it's right in the cavity, right on the eggs. And there's different places, people talk about different places, but that's just, the literature told us that spot, that's where we went. And we had good success with it. So here's a couple pictures. I was talking about photoperiod and water temperature manipulation. So you can see we had a light on these fish. So that light was on a timer. They were in a darkened environment, a darkened tank. And then we also had temperature manipulation going on in that tank as well. And we were able to manipulate those fish. So we had a water chiller to run them through that cold period. One of the weird things we found out was a lot of people told us the fish had to be really cold, colder than we could get some of our fish before, and they wouldn't set their eggs. But we actually found out that they'll set their eggs even with our well water. If we're holding them in warm water and then we take them to our well water, so our well water is about 7.6, 7.8 degrees centigrade. And then we usually raise the fish warmer than that. And if we just drop them into that well water, they actually will set their eggs. And both perch and walleyes do that, and they'll spawn. And so for a couple years, we ran pipes out to our ponds in the winter. We shoveled snow into tanks. We had chillers set up. I mean, we did a lot of extra work to try to get that colder temperature. And then one year we just didn't do it, and the fish spawned anyways. We're like, what the heck, you know? So we got a bunch of long plastic piping for sale too, if anybody wants. Miles and miles of it. But so wild broodstock collection is going out into the lakes, the rivers, the ponds, things like that, where you're going to catch fish with Fightnets, hoop nets, and maybe even hook and line, like the bottom picture there through the ice. A few years ago, I went down and fished the Mississippi River behind one of the dams for saugers through the ice. And then we actually worked with those live broodstock. We brought them back to the hatchery and used them in the spring. Sometimes we partner up with folks like the state. So the bottom right are a couple state biologists here from the Bayfield location, and they're actually netting out in the bay. Sometimes we set our own Fightnets, and these are a couple NADF technicians. This is right down in Ashland next to the big paper mill plant that is sitting there, the Excel powerhouse plant. And those are the kind of fish that we get out of that location. There's really big fish in Chequamegon Bay. It has a really nice population of large walleyes. They're really old. They're about 20 to 30 years old. And they're big. That Fightnet was so full of fish, we had to actually let fish out. And so if you haven't used a Fightnet, the way it works is that the fish lead into it, and they end up in this bottom pot where the smiling guy is. He's so happy he caught all these big fish that day. And that net was actually like swimming away. There were so many fish in it. And big fish, but the problem is that natural reproduction is pretty limited because there's a lot of issues with the bay. So I'm going to talk a little bit about after you've got your fish, so whether it's captive broodstock or whether it's wild broodstock, you've got them in hand, what do I do? So if it's a gravid female, if it's ready to run, the eggs will come out with slight pressure. You just cock the tail back and you apply some pressure and she'll release her eggs. If you force the fish to release their eggs, they're probably not ready and you're not going to have good spawning success, and you got to hold that fish a little longer or find another fish. So when they're ready to spawn, they'll release their eggs. We wipe them down with a towel, try to minimize any water. If you get any water in there, the egg becomes activated basically and it closes up, and you don't have a lot of time to deal with that. And so you got to keep them dry, and you got to be really careful about that, because if you get any water in there, you will have poor success. And we're pretty good, we take a lot of care. We don't spawn a lot of fish to get our eggs. We wipe them down, we're really careful. I've spawned with the state and the tribes and the feds, and sometimes they're just shucking and jiving, and it's like they see me spawn a fish and they're like, geez, what is this? But we have 85 to 90% fertilization rate too. So we're pretty proud of that. And we don't need a lot of eggs, so we take our time and do a good job. So no water with the eggs. You're carefully just spawning that fish down. How many of you have spawned walleyes or fish? Gordy? Other fish or just walleyes? Atlantics, sure. Muskies. Muskies are fun. So it's that, you guys kind of know that procedure. It's just gentle pressure. You can use other things too. I'll talk about that later. So now what happened here by accident is we're adding the male. So that's a male, and you can see the water droplet going in there. And so all of a sudden our dry spawning became wet spawning. So we all of a sudden had to move quickly. And so this was kind of not a really controlled environment. We're out in the lake, we're working, and so now you add the milt, and then you have about 12 seconds to add water. Once the water's added, you have about 12 seconds and the milt is done. So it's a very quick process. So we've talked to people before about spawning when they haven't had good success. It's because they're not doing things in the right steps. So you got to be cognitive of that. If you're in a hatchery setting, you have a little more control. We can actually spawn into a petri dish, maybe collect a couple males, and then apply a couple males onto those eggs. And even in the wild, we always add a couple males because you don't know which males are really viable and which males aren't. And so we're just adding a couple males to at least one female. And sometimes you may even pool your females. It just depends on what you're trying to do. Then you're adding fresh water. You give them a stir. You don't have to wait very long, just a minute or two. The milt will have penetrated the egg, fertilized the egg has closed, everything's done. Then you start to add clay. So we add bentonite clay. So sticky eggs like walleye eggs, you have to add bentonite clay, or you can rinse them off with, what's the other thing? You can rinse them with something. There's a chemical you can add, some people, tannic acid or something. Yeah, and it'll take away the stickiness, but I like clay. We've used clay for 30 years, and it's been used for hundreds of years probably. So you add the clay, it's like a thick slurry or a thick milkshake, and then you just stir it around. A lot of people use a turkey feather. That's kind of the old way of doing it, but paintbrush works too. If you use turkey feather, you got to go shoot turkey. So.
Are you buying that clay?
Yeah, it's just well drillers use it a lot. They use it to seal wells and things, and I think we just buy it at the local ag station. And once you buy a bucket of it, it'll last you forever. So then you're stirring them up, you're coating them with that, and then you would clean those eggs off. You would decant that clay kind of off of them. You add fresh water. With biosecurity things in place, you got to have fresh water that's like well water. A lot of times, we used to just use water from the lake or water from the stream, but now we do everything with well water that we bring from our hatchery. So I carted water down to the Mississippi River, and we rinse everything with our water, trying to minimize any biosecurity risks, because you just don't want to bring that into your facility. And so you clean the eggs off, and then we will give them after they water harden, which takes a few hours, we will then put them in an Iota bath for 100 parts per million for 15 minutes. And so we have used different kinds of Iota. This is called Argentine, but there's other kinds out there. And once those eggs have went through that bath, then they're fairly disinfected, and you can bring them into your facility. And that's something that if you guys are building a facility, you got to think about, is that when you bring in live fish, if you bring in fingerlings or larger fish, you're bringing in whatever the previous hatchery or the previous place had. And that's been one of the problems we've had in a lot of facilities, is that when they get pond raised fish that are feed trained and they bring them into their recirculating aquaculture systems, their systems usually fail because they're bringing in all those bugs. So with eggs, you kind of have a chance to kill all that, and this is even successful for VHS. So this is standard protocol for VHS. So the eggs are then placed into some bell jars. This is our bell jar incubation rack, which you'll see up at NADF. And let's see if I got a better picture. There's a whole picture of the whole complex. And so we've got a rack of jars. Those jars receive well water from up above, and the water is heated so we can get the temperature that we want. We have a series of boilers and stuff that heat the water to where we want it. And then the water goes down through the jar, and it causes the eggs to roll a little bit, so it comes up from underneath. And you want a slight roll on those eggs. You don't want to roll them too hard in the beginning, because if you roll them too hard, you'll kill them. And then later, if you roll them too little, you'll kill them. So the idea is that it's kind of a self-cleaning jar. The eggs roll in there, and as they start to develop, the heavier developed eggs stay to the bottom, and the dead ones, which turn white, go to the top, and you can siphon those out with a siphon tube. So if you ever had to siphon gas when you were a kid or something, now it comes in handy. And you just got to be careful. It's always funny, people who've never siphoned before, it's really a hoot to watch them siphon eggs, because they usually suck too much and they get a bunch of dead eggs, and it's really cool. So yeah, but they taste awful actually. They're not like caviar. So the eggs are in there incubating, and then after they start to develop, actually before they start to develop, sorry, I got to back up a minute, we treat with formalin to control fungus. So even though you're sucking out a lot of the dead eggs, sometimes you'll get a little fungus in there. And so we use a formalin dip treatment. We use a chicken water, and we drill a little hole in that chicken water. I think that's on our webpage, how to do that. And we have a 15-minute drip. Anywhere from 1,200 to 1,600 parts per million is sufficient. And it's based on your water flow. So you have to calculate how much water you're putting into your incubator and what's going through those jars. And that's how you determine the treatment. But it's a 15-minute drip treatment at that part per million. And you don't want to treat them I never treat right after spawning, and I never treat right before hatching. So there's kind of a window in there that you got to watch. And so you got to be looking at your fish too. You can see the chicken water up on top of the bell jar system there. So it just drips into the head tank and it treats that whole tank. And you'll see all this too when we go to NADF. So if you look closely at these eggs, it's hard to tell because it's so dark, but they're so dark because they have their eyes are dark now, so they're eyed up, they're hard-eyed, and they're close to hatch. So when they become like that, we would stop treating at this point. We wouldn't treat anymore. And if you notice, I mean, these eggs look pretty good. I mean, there's some dead ones in there mixed in, but not too many. And most of them have been siphoned off and they're ready to hatch. So when they hatch, let me go back here. When they hatch, they go down into the trough right behind the jar, and then they travel down these pipes and they go into different tanks. And then these tanks have inserts in them. So this is a 4-foot tank with a little shallow insert. These are custom designed. So all this equipment is stuff that we've pretty much designed, and we've worked with the companies, but they now are selling them. You can call Gemini Tanks and get those tanks. And so it's an insert tank that fits inside, and then on top in that tank there's a screen. That's what I want to talk about here. So we got a screen box that unfortunately that one is pretty old. I built that probably 20 years ago or 17 years ago when the facility was first built. And I would like to see steel or metal or stainless there instead of wood, but it works. That screen box has really fine micron screen to keep the fry from going down the drain. And then you notice there's this black air hose around the base. That's bubbling air, and that keeps the screen clean. And if you don't do those things, your screens will plug, and then you'll come in the next day and all your fish are on the floor and you're out of business. So you gotta maximize your screen area and make it bigger, and you gotta have a way to clean it overnight when you're not there. Everybody understand that? Yes?
Okay, maybe a dumb question. Is the hose inside or outside the screen?
It's outside the screen. Yep, and it just goes around it, and then the air's bubbled up. So the screen, the air actually cleans the screen, keeps the stuff from settling on the screen. Because when the fish are hatching, there's all these eggshells and things that are getting washed in there as well. And so those would get on your screen, and they'll very quickly plug your screen. That's a little better picture. You can see the whole setup there. And also, if you notice, we have a light source. So this light source, as the fry start to go in there, the fry are photopositive, and they're going to be attracted to that light source. So that just makes it a little easier for us to collect strong swimming fry. So what we're looking for is we want to collect fry that are 3 to 5 days old. Their yolk sacs are being absorbed. They're strong swimming. They swim to the light. I don't want fry that are back over here in the dark. They're just kind of like laying there half dead. Those fish are not going to perform for me. So getting eggs and getting fry for us is not a problem. We have millions, literally, of them over there right now. And if I only need a few 100,000 of those, I want the best performers I can get. So that's what that light source does for us. I'm going to switch gears for just a minute, because walleyes aren't the only fish, right? There's this other fish, this yellow perch. And so again, you got to have broodstock, right? So you got to get your broodstock from somewhere. There's different sources for broodstock. And then you got to go through spawning that broodstock. And some of the same things we talked about with the walleye are true for perch as well. You can use out-of-season spawning. You can use hormone, and you can hand spawn them. You can let them spawn in a pond and then collect the egg strands. So there's different ways to do. We have a really nice video that was put together a few years ago by Jim Held, who was one of our extension agents, and I don't know if that video is available very easily. You have to buy it, but it's a really good video that talks all about perch spawning, and he goes through the whole thing and does a great job of it. So if you're interested in perch, get that video, and we can help set you up if you need to find it. Emma can. So when you got your strands, so yellow perch, I don't know if you can hardly see them in here, but there's strands of eggs. So yellow perch eggs are in a matrix. So that matrix is like this gelatinous thing. It looks like frog eggs. And sometimes you look in a lake or a pond and you might be thinking you're looking at frog eggs, and really you're looking at perch eggs. So when they come out of the female, there's this long gelatinous matrix, and it almost looks like an accordion. And you can stretch it out and you can lay it on this grid work. So that this is what we do at NADF, is we lay it on this grid work, which is made from lights. Those are the light grids. So you also find out when you work at a hatchery that you find all sorts of interesting uses for things that you never thought possible. And so those are light grids that we buy at an electric warehouse. And we lay them in the tanks, and then we put the strands on there, we stretch them out, and we put ties. Those are plastic zip ties just to kind of hold them in place. You won't see yellow perch eggs at NADF right now because we're not spawning yellow perch anymore. But I wanted to talk about it a little. There's a better picture showing a strand. And there's like 1 million perch eggs in there. They're smaller than walleye eggs. But you notice it's a whole different thing. They're not individual. They're in this matrix. So you're incubating them in here. You can also incubate them in like a horse trough or a rectangular tank by building this chicken wire support, and then you drape the eggs on it, and as the water goes through the raceway or the tank and keeps them well oxygenated and taken care of. So yellow perch are a little different, so you have to look at them in this matrix. And here's a baby perch, and you can notice, you can see its eyes, you can see its yolk sac, you can see its GasBladder, or, well, what is that? I don't even know what that is. So you can see this fish, but it's not ready to hatch yet. It's not dark enough yet. So you got to look at them in the matrix. And then when they get ready to hatch, this is what they look like. So you notice the eyes are fully pigmented. It's got fin buds. The yolk sac has elongated a little bit. And so that fish is ready to hatch. So at this point, we pull the matrix out. We siphon the matrix into a bucket. And we take a drill-powered paint stirrer, and we beat the heck out of them. And we release those fish from that matrix. And you think it's going to hurt them, and you think it's going to kill them, but they can handle it. And if you think about this in nature, what's happening in nature, those eggs are, when this when they get ready to hatch, the matrix is breaking down, there's waves, and there's wind, and it's a little turbulent area out there in the pond, and it would break that matrix up. So we're simulating that same type of activity to release those eggs. So now, once they're released, now you got baby fry. So whether you have walleye fry or perch fry, you've got to enumerate them somehow before you put them into your pond or into your tank or what have you. Because if you don't enumerate, if you just dump a bunch of fish in, how do you know what to feed them? How do you know what's going on? How do you know your success? You don't know anything. So we have really looked at this pretty hard because we used to do a method. We used to do this method, fry estimate by hand counting. So we're thinking we might have you guys all help us count that 1.5 million fry tomorrow this way, or today. We'll see. I don't know, how many days do we have for the workshop? So if you were to enumerate this way, and by the way, this is one of the only ways it works for perch, because you can't run them through our counters. So that's been a problem for us. But you would put your fry into a bucket of water or whatever, you mix them all up, you take a sample, that's a graduated sample in that container, you know exactly how much you have. You count the fry. So this is a really great project for school kids, right? And technicians and interns. Because you got to count like 500 fry, and they're the size of your eyelash and sometimes smaller. So then you can kind of guesstimate how many you think you have in that bucket. And I guarantee you it is a guesstimate. We've looked at it and we found that, I mean, you can be off by 50%. So for us, that's not acceptable. So we use a gen sorter fry counter. You're gonna see that in action today. We're gonna do this today because we have fish. So remember, we're a working facility, and Kendall's up there taking care of the fish, but the gen sorter fry counter, we put fish in there, it counts them with a light beam, and so we can get pretty accurate counts, probably within 5%. And then we've been playing around with this expert fry counter. This is something that's made up in Quebec, and that you put fish in this bucket and you put this top on, and it's a computer, and you push a button and it's supposed to tell you how many fish are in there. That's how it's supposed to work. Unfortunately, it's not that easy. And so we'll talk about that a little bit later. That's only, I forget how much those were, 5,000? Yeah, pretty cheap. Maybe we might sell you guys one for 4,000 because they're pretty accurate, but we've run into some problems with them with our species. One of the problems is nobody knows anything about walleyes. So we're developing the protocol. The gen sorter is still, it's about 5%, 95% accuracy, 5% either side, and it's fairly expensive, but you can rent it. So we rent ours for $250 a year, or you can buy one for about $7,500. To me, it makes sense to rent. And then I send it back when we break it, and they fix it. So I'm coming to the end. Ooh, just in time. You're ready to go. So Emma's going to talk about what do you do with these fry after you've enumerated them. And I just wanted to leave you, this is a little ant down here, and he's dragging a spider or a fly, I think. And at first I saw this thing out. This was at the facility one day. I was walking out and I saw this little guy, and I was like, wow, I was pretty impressed. You know, I'm like, he's taking on this big fly he's gonna eat for weeks. And I was like, never give up. But then I started thinking about it, and I'm like, or did he bite off more than he can handle? Sometimes we feel that way in larval culture, so I think that's it. Yes.
So this presentation is on our intensive walleye larval system. You'll see a lot more of this. You'll actually be working in this system with Kendall today, so you'll learn kind of how we clean, how we sample our fry in the system. There's been increasingly a lot of interest in raising walleye intensively, and we'll talk about the benefits of that, especially to produce a fingerling that is feed trained. So we'll talk about it throughout this presentation, but walleye are a highly cannibalistic and carnivore species. They want to eat something living. So we're basically training them to eat a pellet throughout this larval period in our intensive system. So we talk about intensive versus extensive. So intensive means indoors. They never see outside. We don't even start them on live feed. It's all commercial formulated feed, all indoors. So these are very biosecure fish. They never see outside. We're controlling everything from temperature, water quality. We try to control things like cannibalism, GasBladder inflation, but we'll talk more about that throughout this presentation. So no matter what fish you are working with, you really have to go back to the biology of the fish. You really have to understand the fish that you're working with. So for walleye, there's some definite key points at this larval stage that we are looking at. For one, the size. These are very small. Greg mentioned they are the size of your eyelash, so they're about 6 to 9 millimeters as sac fry. So sac fry means they have their yolk sac, they have their oil globule. That's their food source for a period of time until they start to look for an external food source. Greg also mentioned that they are photopositive in behavior, and I'll show you a video on that. 3 to 5 days only for exogenous feeding. That means that 3 to 5 days that yolk sac is absorbed and they're looking for an external food source. So that's formulated feed that we're feeding them. They'll be looking for that in about 3 to 5 days. And also cannibalism starts immediately at that exogenous stage. So the soonest that they can eat something external, they're going to be looking at each other and potentially cannibalizing. So some people think that that doesn't happen for several weeks after they hatch, and we've seen it immediate. So that's really important to note. And there's two kind of developmental stages. Of course, feed acceptance is very crucial for that initial survival. You want to make sure that you're feeding them a palatable feed that they're going to accept, that has the correct size, correct formulations, and also GasBladder inflation, and we'll talk about what that means, but that's a crucial stage for that fish to survive or have correct growth or best fitness. So I have a size comparison here for you. So here's an Atlantic salmon fry. They're quite large comparatively to our cool water fish, but this guy here has been absorbing his yolk sac. He's about 25 millimeters in length, so over 2 centimeters. This is an Arctic char. This is about 24 hours post-hatch, and he's absorbing his yolk sac, and they're quite a bit smaller than other cold water fish. This one is about 14 millimeters in length. And then our walleye, or perch, are much smaller. So we're talking 6 to 9 millimeters when they first hatch out, and their development is quite quickly, but it's still, you're working with a very small fry. So we talked about photopositive activity. So they are attracted to light and light-colored tanks, a light source. Do you guys know why that is?
Get to the top of the water, it's like their gas bladder?
Good guess, maybe potentially, but I don't think that's the main reason. You guys remember what they eat when they initially hatch out? Plankton, yeah. So zooplankton is their initial food source. So they have this innate behavior to be attracted to light because zooplankton are near the surface of the water eating phytoplankton. So they have an innate behavior, even though they still have their gasbladder or their yolk sac absorbing, that's their food source. They are just innately attracted to light, light-colored tanks. So this is that picture. We've got that insert tank. So these are immediate after they hatch out of the bell jars. They come down into this insert tank. We have that light set up, and we're getting those strong swimming fry, excuse me. So this is a video of what that looks like. So again, we're working with millions of fry here, and they're all concentrated. Those strong swimming ones are concentrated near that light source. So again, we scoop up those fry. Those are the ones that are going to be surviving, or in the strongest of that millions of fry we hatch out. We have any kind of container, so it depends on how we are kind of enumerating them, but we could scoop them up with just a normal container, anything, and then pour them through our larval counter. So we don't net them. They're very fragile and they're delicate, so we always want to use water when we're moving them at this stage. So 1 to 4 days of endogenous feeding, that means they're absorbing their yolk sac, so they're internally using that food source. So this is a very newly hatched out fry. They look really alien-like. There's not a lot of development here. You can't really see anything in their digestive tract, and they really don't even have a mouth at this stage. All they have is that photoreceptor. They're just looking for light, so really underdeveloped looking. But quickly, in about 2 to 3 days, they'll get that mouth formed. They'll begin to open that mouth. This one has not accepted feed yet, but they're still absorbing that yolk sac. This round kind of circle there, that's their oil globule. That's also a nutritional source for them, so they don't starve. They have another couple days to look for feed, but you can see the digestive tract forming there too. And they're actually developing teeth at this stage, which is pretty crazy. So then just in another few days, they're able to accept feed. This is exogenous feeding now. So really, we have 3 to 5 days to get feed to these fish. So if we don't get feed to them, they can either starve or they can cannibalize. So these fish are only about 12 days old, and honestly, they're about the same size, and they just cannot help themselves. They want to eat something living and moving, and that's a lot of times that's each other. So we call this, sometimes we see unobserved mortality, which means the entire time that we're raising these fish in the larval system, we're counting mortalities. Every day we're counting actual mortalities, dead fish in our tank. At the end of that 30, 40 days, we drain the tank and we count how many fish we have. And there's this big gap in numbers. There's this big missing fish number, and we call that unobserved mortality, and a lot of that is cannibalism. Those fish had gotten eaten out of that tank. So these 2 key developmental stages, of course, feed acceptance. We can track that, we check that, make sure our fish are feeding the feed that we're providing them. And also something called GasBladder Inflation, and that is what this little air bubble is here. So for species like walleye and perch, they have to physically go to the surface and inflate their gas bladder, like you had mentioned. They have to come up to the surface, grab basically a bubble of air and swallow it to inflate their gas bladder. Cold water fish like salmon, trout, char actually use their blood to inflate that. They don't have to physically come up to the surface and inflate that. So this is a key step in their development. So there's a specific interval that they have to do this in, so it's about 5 to 12 days. If they wait too long or are unable to do it, they will not be able to inflate that, and they will never be able to inflate that after that interval stage. So these fish here are about 10 days post-hatch, and most of them have inflated their gas bladder except for one, and that's right in the middle here. So maybe he has a couple more days to do that, and if he cannot, or she, I keep saying he, they will survive, but we see increased issues with non-inflation of gas bladder. Going back to the cannibalism picture, you notice that this fish here does not have a gas bladder. So gas bladders help the buoyancy in the fish. It helps their swimming ability. And if they cannot inflate it, they cannot swim correctly. They're poor swimmers, so they have increased cannibalism or predation on them. And they also can cause deformities. We did a project with yellow perch looking at gas bladder inflation was one of them, and we see a correlation between these spinal deformities and being able to inflate their gas bladder. We're not quite sure whether one came before the other, if they were spinally deformed and they couldn't inflate their gas bladder, but there seemed to be a correlation between these two things. And you can see the bottom picture, that's a small fingerling, so they will survive, but some of them look like an accordion, they're so spinally deformed. So you really can't use that fish at all. And if that fish was a walleye, most likely it would be cannibalized on. So getting to our walleye larval rearing room, so you'll see these rooms at the facility. This one is kind of our older room. We also have a newer room. So this one has 19 tanks. So it's a very intensive system. So it takes about 8 to 10 man-hours a day to do cleanings in this, feedings, sampling. It's very intensive for our staff. So these fish are in the system for about 30 to 40 days, so a little over a month. So the idea is to get this fish on from a kind of intensive formulated feed up to a 1 millimeter pellet. So when they come out of here, we're feeding them a commercial 1 millimeter pellet, like a spreading diet, what else would we feed them besides spreading? Europa is what we feed them. No, when they come out. Yeah. So we want to get them on a commercial diet, basically. So this room is really intensive for feed training. And this entire system, the design and the setup is basically designed with the key points that we just talked about in mind. So why do we do intensive rearing? We talked about a lot of this already, but you can have increased monitoring and control, right? So you're controlling temperature, we're monitoring for water quality, growth. We're giving them a commercialized formulated feed, so the quantity of the feed, we know exactly what we're feeding them every day, and they have very good quality of feed as well too. So this has a high protein diet, and of course because of these reasons we see increased growth and survival in these fish. So like the picture shows, the top picture here is an intensively reared fish in our system, and then the bottom is our pond reared fish. So generally we see this, we get a better fitness, better growth with those intensive fish. You can manipulate growth of these fish, mainly at a fingerling stage after you get them out of these systems. You can slow the temperature, slow the growth by lowering the temperature. And if you have a group of fish that is ready to go, ready to market, and you want to slow a portion of those down to help meet your production schedule, you can lower that temperature and they'll kind of lower their feed rates and lower their growth. We did a project last year looking at cold banking of walleyes, and we're not quite sure if they'll come up to that full growth like they were before we cold banked them, but we basically put them through a winter period and their growth pretty much stopped. And we were going to see if we brought them back up to the worm temperature, if they would overcompensate for that growth or get back on their schedule before we put them on that. We're still doing a few research on that, but it's an option. And Greg mentioned we can utilize out-of-season spawning of our broodstock. So if you have an intensive system, you could be able to manipulate your broodstock to give you fry multiple times a year instead of just once in the spring using a pond system. And lastly, there's a large market for intensive feed-trained fish. So these fish are biosecure. They are on a pelleted diet. So for aquaponics systems, people are looking at this, and for intensive research systems as well. So just getting into a little detail of the tanks themselves. So for one, you look in this tank and you immediately notice that it's very cloudy or turbid. So we utilize turbid water using a bentonite clay. So there's a specific clay that we use from Kentucky that's actually very smooth, so it doesn't affect their gills. But it does a lot for the larval fish because, for one, it limits clinging behavior. So what that is, I've got a video here. So this is showing a video of a clear tank without turbid water. And you'll see that the fish actually cling to the side of the tank and they aren't dispersing across that tank's width. So the issue is that the light source above the tank comes down and reflects off the side of the tank, so they see that reflection and they're attracted to that light. So unfortunately, this causes starvation because they aren't looking for their food. They think that it's on that tank wall. So having the clay in the water kind of refracts that light, causes it not to bounce off the side of the tank walls. Turbidity also limits cannibalism, so if they can't see each other as well, they're not going to be cannibalizing as often. And there has been research in the Walleye Culture Manual that having that contrast to that feed is better feed acceptance. So having turbid water provides a contrast to that real orange colored pellet, and they accept that feed better. This is just a few diagrams and showing how this is working. So in each diagram, on the left side are the clear tanks, and on the right side are tanks with the turbid water. So in this first picture A, the light is just going through the water column and bouncing off the side of the tanks, causing the fry on the left side on the second picture to kind of concentrate on that tank wall and not disperse and not look for feed. And then in comparison, the top picture here, sorry, I need a laser. The top picture on the right side is the one using clay, so that light source reflects off and the fry disperse better throughout the water column. Yes?
These benefits, they hold true for perch and other species?
We looked at perch, and we looked at turbidity and color, tank color and spray bar, which I'll talk about in a minute. And I believe they did better with the clay water, but perch we weren't as successful raising perch in these systems as we are with walleye. They're just not fit for what we're doing with the walleye systems right now. But for perch, they tended to do better in the turbid water with the black tanks. They just weren't as successful. So there's still a couple missing links with perch, but I don't know if we've tested any other cool water species in these tanks, just walleye and perch, right? But in cold water species, we don't do this at all. We do something completely different that you'll see at the facility. So tank color also is important, and it works together with turbidity to help disperse those fry. So again, you want that black tank color. It helps not to reflect that light as much. And then we do paint the bottoms gray because we want to pull the fry down into the water column as well too. So we want to keep it kind of light down there. Because they're still attracted to light colors, we want to kind of bring them down into that tank as well too. So moving on here, we have 24-hour feeders. So we want these fry to be feeding consistently, 24 hours a day. We don't want them to go hungry. We want them to continue growing throughout the day and the night. So these are feeders that we have built at the facility. These are using a mechanical time switch, actually. So you can buy these micro feeders, they're called, online for a couple $100, or you can make them yourself for about $60. We were able to do that. These steps are on our webpage, but basically it's a mechanical time switch with a dial, and we built or cut out kind of a plexiglass dial that slowly turns. So in 24 hours, that dial will have turned and the feed slowly drops off into that tank. It's hard to show you with pictures, but you'll be able to see this at the facility. But kind of a much cheaper way of doing this, and it's still pretty effective and efficient. So it's really important to keep up on this growth. This was some data that we took in 2015, looking at really at that day 20, those fish just explode. They are doubling in size every few days. So we really have to make sure that we're sampling those fish to be able to feed them enough feed in that system. So in less than 2 months, they'll be out of the system in about a month, 40 days. But at day 53 here, we're looking at 4-gram fish that's about 80 millimeters or 3 inches. So that's a really nice 3-inch fingerling in less than 2 months. That's feed trained. And I just have some pictures here showing the very quick development of these fish. So the third point here, we've got spray bar, I mentioned that. So what the spray bar is it's a hose basically of water with a nozzle on it, and it's basically misting the surface of that water. And something so simple is very important for things like GasBladder inflation. So you can see in the pictures on the right side, we were looking at, well, does spray bar make a difference for perch like it does in the walleye? The top picture is very clear, very clean water. If without a spray bar, all that oil and that feed sticks on the surface. And we looked at GasBladder inflation rates. With the spray bar, easily over 90% GasBladder inflation, not really an issue. The no spray bar, we saw very poor GasBladder inflation rates. So those fish are so small and weak that they cannot break that surface tension or break through that oil and that feed, and they aren't able to inflate that GasBladder. So it's really important that something is breaking that surface tension and pushing that feed and that oil into the tank. Point 4 here, we have rotational inflow. So even in all of our tanks, in our big tanks, it's really important to have that rotation, kind of that velocity inside of our tank. Especially for our walleyes, it actually reduces cannibalism. So we want all the fish to be facing into the current so they're not, kind of T-boning each other, looking at each other. We want them to face into that current. And how we do that in our larval tanks is pretty simple. It's just a PVC bar basically, or pipe that's capped at the bottom, and we have little holes, you can't see them very well, but little holes drilled down the side, and we're basically pushing that water around in that tank. And we're also maintaining good water quality that way, so all the water is moving around in that tank. There's no pockets of poor oxygen or decay or poor water quality. And we also want that feed to be moving across the water column as well too. One of the last points here, we've got a center screen and a standpipe. So it's easier to see this in a tank with no water, but we basically have a very tall screen, kind of like a tubular structure. And the reason for that is you have enough surface area for that water to leave. So we have a lot of clay, a lot of fungus happening in these tanks, leftover feed that will quickly clog a drain if we just had a bottom drain. So we want increased surface area in that. And then we also have something called standpipes. That we have one standpipe in the middle of that screen and then one on the outside of the tank. And Kendall will talk more about how that works, but basically we're controlling water level and water suction out of that tank. So if we were to lose water, the water couldn't come to this tank, that water level would stay at that standpipe level. So it gives us a little bit of control that way. Just quickly, we utilize 3 different screen sizes in our tanks throughout these 30 to 40 days. We start with a very fine screen, right, because the fry are very small, but then quickly we want to move up in sizes. So we've got a 400 to 1,000 to a 2 millimeter screen. So the soonest we can get to a larger screen size, obviously we want to get the debris out of that tank as quickly as possible, but we just have to wait for the fry to become large enough not to fit through those screens. The last point of these systems is we have an overhead dimmable lighting over each of the tanks. You can see here. So these are dimmable with a unistrut here, a rotostrut, what is it? Rheostat. Rheostat. Yeah, I don't know where I came out with that one. So a rheostat is the dimmable lighting, right? So we want to be able to keep this room nice and dim. So while I enjoy dim lighting, right? Because as they're older, they aren't as photopositive, but we want them to be feeding consistently. When they are photopositive, we will come into this room and turn the lights up so that all the fish come off the bottom so we can clean underneath them. When they turn photonegative, which is in about 2 to 3 weeks, it's evident when they do, we turn the lights on and all the fish dive down. So we want to keep the lights dim at that stage so that they aren't all sitting on the bottom. So we will talk more about that, or Kendall will share more about that in the systems. This is one last video. So when they're photopositive at this stage, you can see them, they're coming up to the top. They're pretty dark colored in their bellies, so it means they're accepting feed. But you can see I'm standing right over the tank and they're not, they're not skittish yet. You can see them eating the feed at the surface. So these are probably a couple weeks old at this point. All right, so just in summary, so intensive larval rearing, not just for walleye, but for any species, you really want to focus back on the biology of the fish. You need to understand what the species you are that you're raising and kind of the key aspects of those fish. So there's technological advancements, especially for intensive systems right now. For formulated feed, we're doing a study this next month on different formulated feeds for larval rearing for walleye and sockeye. There's different management, equipment is always coming out new designs. And there might be new and improved species or strains to look at for intensive rearing. Right now we're comparing walleye and sockeye, but there might be other strains out there, species that do better than others. And not all species are fit for intensive culture at this time, like yellow perch. So we did 3 years of research on yellow perch, and there's a few things missing that we aren't aware of yet or don't fully understand because they're just not as successful at raising intensively as the walleye or the sockeye. So we are really trying to turn our attention to these species because we can do it successfully. We know it can be economical or productive to raise the walleye or sockeye, so we're kind of turning towards that species and trying to look away from the yellow perch for intensive rearing at this time.
Switching gears a little bit, we talked about cool water, and Emma gave a great talk about the larval systems. We're going to switch gears and go into cold water. So we're looking at water temperatures of 45 to 60 degrees Fahrenheit. And again, the fish can go colder and they can go warmer, but those are optimum temperatures for what we're dealing with. The egg incubation times are 30 days and longer. So not like the cool water fish like we talked about a little bit ago. Cold water fish have a much different setup. They have a much larger yolk sac, and they feed off of that for a lot longer time. And they're much larger, as Emma showed that one slide that had all the different sizes is really good. So the species that we deal with or that we have dealt with in the past, and what I'm talking about is Atlantic salmon, Arctic char, brook trout, rainbow trout, brown trout, lake trout, and Pacific salmon, which are your coho and your chinook. And there's other cold water species out there that I'm not talking about, but that's kind of what we're dealing with. So when we talk about domestic, again, broodstock, is it domestic broodstock or is it wild broodstock? So do you have your own broodstock in your hatchery, or do you have a lake with broodstock, or are you going out and collecting it? So regardless, you're collecting those in some way, and we'll talk about that, and I think Mike will talk about that in his presentation as well. So we have artificial spawning and natural spawning. So artificial spawning is where we actually collect those fish. We would use a chemical called MS-222 to knock the fish out, like in a horse trough here, where we have Nate and Kendall. So Nate actually works with Mike now. He used to work for us, and now he works at the state hatchery. So there is life after NADF. Whoa. But Kendall doesn't know that, so don't tell him. So we knock the fish out with MS-222, and then we hand spawn those fish. There's some places I know that use natural spawning where they let the fish spawn and then they go and collect the eggs in a simulated raceway or a stream or something like that. But we don't. So we hold our broodstock. This is our Atlantic salmon broodstock. So some of these fish they're 30 to 50 pounds now, and they are 6-year-olds. You'll get to see these at the facility. We've been spawning them in the fall for several years, and we hand spawn those fish, and they're getting so big now it takes 3 of us to spawn them. You'll see a picture in a minute of one of those fish being spawned. But so when we're talking about broodstock, it's important if you have a broodstock on your facility that you take care of that broodstock. I oftentimes hear people talk about putting their broodstock down at the end of the hatchery, getting the last water, and oh, we feed them once in a while, and they don't take care of them. Your broodstock should probably be in the front, and they should be getting taken care of. They should get the best water, and they should get the best nutrition. This is really important to have good eggs if you're going to have a good program. There's companies out there like Trout Lodge and now Riverence that that's all they do is they raise broodstock, they spawn them, and they sell those eggs to us. I'm sure some of you guys get eggs from Trout Lodge. And so their whole business is taking care of that broodstock, and I guarantee you they're looking at nutrition, and they should be looking at genetics. And so what that means is they're selecting fish that are going to give them the right, the good growth, good fish health. Maybe they like feed better, things like that, things that we are looking for. They're selected for those genetics. And I'm not going to get into genetics and all the crossings and things like that, because that's a whole nother workshop. But there's lots and lots of people that just do genetics, and same thing with nutrition. So make sure your broodstock is getting good nutritious food, and broodstock demand a little different food than fish that you're growing for market or something. They have to have more vitamins and minerals. Sometimes you want you add color to that feed so that the eggs look nice. There's different things going on there, and there's nutritionalists out there that can help you with that. The other thing is that the age of the broodstock. So when you start to spawn a fish, usually as the first couple spawns, the egg quality isn't so good, so you got to be aware of that. But then as they mature, you'll get better egg quality. But then on the other side, the egg quality is going to start to decrease again. So you have to plan for that. If you're going to run a program and you're going to have broodstock, you got to understand those things. Health. So the health of the broodstock is very important, and it relates to everything I just talked about. And it's giving them good environmental conditions as well, and a good place to live. So if I go back to that last slide, those fish have a really nice setup there. They have good clean water, they get all the feed, it's high quality feed, we take really good care of them, and we benefit because we have really good egg production from those fish. I don't know why that picture's in there twice. So when we start to spawn the fish, as I mentioned, so we would knock that fish out with MS-222. So this is some pictures of our in-house lake trout. So you'll get to see some of these fish in our raceways, and we were hand spawning those fish. So we've knocked the fish out. We are gently massaging those eggs out of those fish. So again, you're checking the fish to find out when they're ripe. They're spawning in the fall, and they're spawning over a period of time. This is probably in October, November, right around deer season. And so that's one thing in this business, is that everything revolves around other activities. And the things you love to do sometimes you don't get to do because you got to spawn fish. But so we've removed the eggs from these fish, and then we're adding milt with this eyedropper. So this is a really controlled setting, right? We're making specific crosses with these fish when we were doing this. So it's not always this nice and this neat. Here's an Atlantic salmon being spawned, and so you can see the size of that fish, and you can see it takes 3 of us to spawn the fish. And you got Emma's collecting the eggs, Kendall's applying pressure, Patty's holding the tail. Patty has worked for Riverence out west. She was a egg specialist or whatever. The other thing is you see the green hose, so that's oxygen. So we would inject oxygen into the cavity to help push the eggs out. So that's another way of spawning those fish. It helps, because honestly, trying to massage all the eggs out of these fish, you get tired, and your hands get tired. And I mean, we're pushing with our fists sometimes to get all the eggs out. And it's important to get the eggs out of the fish, because if you leave them in there, they cause problems for the next year. But so these fish now are so big, they're just too big to spawn anymore. So we're finishing up that project. But so once you have your eggs, so you fertilize them, so it's very similar to what we saw with the walleye videos I showed you, except we don't use clay. We just collect those eggs, and then we would iodine them, just like I talked about before, 100 parts per million for 15 minutes, and then we would place them into an incubator. This is a picture of a vertical tray incubator, or a heath tray. Some of you heard of a heath tray, hopefully, if you're doing cold water fish. There's other types of incubation units out there. There's the Montana hatching box. There's hatching baskets and troughs. I've seen those in facilities where the water kind of upwells underneath them. There's the bell jar incubators that we use for the walleyes. There's big red incubators, which are these little tubes, which are kind of a joke, but some people may have seen those before. And then there's cannon tube incubators, which are big pieces of PVC pipe where you run it's kind of like a bell jar incubator, but it's a flat bottom. So there's a few differences with cold water fish and how you incubate them and how you go through that process and how you roll them. You got to be careful about that. So we use the heath trays because we like them, and you'll get to see those. I don't have any eggs to show you right now, but so we talked about iodine treatments, and here we are iodining the eggs. So we always iodine our eggs when they leave the facility, and we always iodine our eggs when they come into the facility. So one thing I don't want to be blamed for is sending a disease to somebody, and one thing I don't want to get is a disease from somebody. So, and no matter what, I have people tell me, well, we've iodined those eggs before they left our facility. I still iodine my own. I want to see those eggs in the iodine every time. I've heard of too many problems where somebody thought they were getting disinfected eggs and they didn't get disinfected eggs, and all of a sudden their hatchery got shut down because they brought something in. So, and these pictures also show, so if you notice there's a temperature monitor. We're monitoring the temperature and the oxygen in that iodine disinfection there, and there's also some ice in there. So that was probably some eggs coming in, and we're matching the temperature, and we're slowly tempering those eggs before we put them on our water. So it's really important to take your time and go through these processes, and a lot of times those eggs might not come to you till late in the day, or you're busy, you're trying to get out and go walk your dog or whatever. Stay and take care of your eggs, temper them in, don't rush the fish, because a lot of times you'll kill those eggs if you're not careful. So, and you see this package on the upper right, that is a styrofoam insulating box. That's how the eggs come. And sometimes they wrap them with cheesecloth, a little wet cheesecloth, but eggs can be shipped dry, basically like that, just moist. They don't have to be in water. You can also ship them in water. We just got walleye eggs from Iowa that were in water, water and oxygen. But you can ship them dry, and they will show up FedEx overnight, and they'll be good to go. The nice thing about dealing with some of these companies too that do broodstock is that you can get eggs anytime, any day of the year almost. You can get eggs. So if you're ordering rainbow trout eggs from Trout Lodge, you can say, I want eggs in March, and I want eggs in June, and I want eggs in August, and you can get them. So I don't know. You have to talk to the companies that have the fish to find out what's available. I'm not sure if brook trout are available through Trout Lodge. There's some companies on the East Coast that might have brook trout.
I guess the question was, any day of the year?
If
look at the cold water.
I don't, yeah. So it depends on the species. So rainbow trout, there's enough demand. They have broodstock that's producing eggs all the time. I don't know if there's enough demand for brook trout, so I don't know if that would be true. Atlantic salmon right now, you can only get eggs in the fall, but they're working on getting them around the year. Coho, I think you can get them a couple times a year right now.
But as a species, you can year-round.
You can trick them. You can still trick them. Yep. So this is the eggs getting put into a heath tray, so you can kind of see how that looks. And the water upwells underneath that, so it's a lot of fresh water going through the heath trays. The young man there working now is the fish manager for Urban Organics, Lee. He was at the WAA conference giving a talk. And so he started with us as I think he was a summer intern. Now he's running one of the biggest aquaponics facilities in the world. So you can't read this really well probably, but I just wanted to make a note of egg treatments. So we do treat our eggs and the heath trays as well. So we pick them by hand and we treat them with chemical. So this is a treatment that we use that the Atlantic salmon folks wanted us to use. So we treated with iodine some days, and then we treat with formalin on other days. And then it's a 20-minute treatment period. So this one, that big barrel is a barrel of formalin, and we're injecting that right into the water that goes into the heath trays. So we don't actually have to touch it. We just turn the dial and walk away a little bit, but you never leave during the treatment because you never know what's going to happen. So that's just more pictures. The other thing is on the right, here's the old chicken waterer again. You don't need a big fancy timer and a big fancy injection unit. You can treat by hand with a chicken waterer, and that's been done for a long time, and it works just as well. So you don't have to be all fancy. You can do it the old-fashioned way. And those egg trays hold a lot of eggs. So for most facilities, you're not going to have tons and tons of egg trays. You're going to have one stack maybe, or a half a stack, and that's it. So factors affecting egg development. So we have a lot of things affect egg development. And remember, these cold water species, the eggs take a longer time to incubate. So they're sitting there. So you got to take care of them. So light is important. So we keep our eggs in the dark most of the time unless we're looking at them. Water temperature affects it's TUs, so that's how temperature units, so that's how quickly they hatch. And so if your water temperature is colder, it takes a longer time to hatch than if it's warmer. I'll talk a little more about that in a minute. Oxygen is really, really important, so you got to have well oxygenated water. You also, along with that, you want to be careful of not having any chemicals or bad things in your water like iron or too much nitrogen. Nitrogen's a problem as well. Nitrite can be a problem as the eggs near hatching. They start to metabolize a lot of stuff, and the egg trays can fill up with that. So you got to make sure you have lots of fresh water running through your system, and good water. So talk a little bit about temperature units. So a temperature unit, a daily temperature unit, DTU, it's 1 degree Fahrenheit above freezing for a 24-hour period. So if you have 40-degree water for a day, that's 8 temperature units. And so different species require different temperature units. So we use this both for cool water fish and cold water fish. And these are just some examples. So estimated daily temperature units for rainbow trout, 550 to 650. So depending on the temperature of your water, you can kind of calculate out when are my fish going to hatch. Look at the lake trout all the way up to 880. And this isn't set in stone, so don't say, well, you told me 550 and my eggs hatched at 530 and I didn't have feed, so they died. This is just a general thing. Notice the warm water or cool water fish like the walleye, it's a little lower on the temperature unit. So like I mentioned, they hatch faster, plus the water you're raising them in is a lot warmer. Look at northern pike, only 180. So you don't have a lot of time. So when you've got those eggs, those fish are going to hatch, and you got to be aware of that. So these are newly hatched. That's a newly hatched fry. So remember Emma's picture. This is a pretty good sized fish, or fry, compared to a walleye. Has a large egg sac. It's 20 millimeters long. A walleye is about 8 to 10. Perch are about 5 to 6. Lake herring are about 4. So you can go from there, and so look at that big egg sac though. So that really helps keep them alive. And so we put them in the heath tray incubator system. They hatch. They stay in there for a little while. They've hatched out. These fry have absorbed their yolk sac. Again, we would enumerate them. So here we are weighing the fry that are coming out of the heath trays, and we're putting them into an insert tank for initial rearing. So we would weigh those fry, we would count out a series of fry, and then we would weigh the whole bunch of them, and then we figure out how many fry we think we got. And we're always right, because if we're not, we tell somebody to count them. Good luck. This is a little note I wanted to make about shipping fry. So oftentimes fry get shipped places, and this is how we package them up, whether they're walleye fry or they're trout fry or salmon fry. We package them up in bags with a little bit of water and a lot of oxygen, pure oxygen, and then we put them in coolers, and those can be shipped or maybe taken out to a lake to be stocked. If you're going to go right to a lake, it's just an easy way to move them around. These are the insert tanks again. So they're 4-foot insert tanks, 4-foot round fiberglass tanks with inserts that drop in. And we use them for a lot of different things because they're really easy to work in. And you'll notice that when you get into the facility, you'll see those. The fry are right there, they're right in your face. So you can see them, you can work with them, and you can notice if there's any problems. If you put them into a deep tank and they're way down at the bottom of that tank, they tend to get neglected, and you don't always see everything down at the bottom, what's happening. So we use these insert tanks a lot. That's just a picture of a bunch of fry or fingerlings. And actually, if you look at those fish, they're in really good shape. They've got good fins. There's still quite a bit of tank space around those fish, so the density should be okay. Some of these fish like to be put together. They like to school together, and if you put not enough fish into a tank, and they'll spread out, and you'll get bullies in the tank, and they'll pick on each other. So sometimes it's good to force them to be together. So when we first start feeding them in those tanks, we're feeding them every hour. We've got this really fancy time clock that we developed. I'll give you the designs for that for $100, and you can build your own. But you know, one thing when you have a bunch of people working in a place, we're like, did the fry get fed? We don't, instead of trying to find somebody, we just have this simple time clock. So we're starting out with small feed. It's like a mash like Emma was talking about, or size 0 to 1. And then we're feeding them, and we're cleaning a lot. We're making sure those tanks stay clean. So we're cleaning morning and night. Sometimes we even clean in the middle of the day. We try not to overfeed, but you want to give them enough feed. So we're usually at about 5% for total body weight to feed per day to start. And a lot of times that's overfeeding. So that's why we clean a lot. But you got to kind of match that. If you don't, if you let it go and you let the feed build up in the tanks, you're going to have problems with bacterial gill disease. You might even have issues with cold water disease. There's different environmental things that are caused by poor hatchery operations. And it's stuff that you're doing, so you can fix that. So we do use salt a lot when we're treating for any of those gill issues. Fish like salt. And these cold water fish, these rainbow trout, brook trout, all those fish, giving them a salt dip, it's our first order of business if there's a problem. And then also there's treatments with peroxide, and there's treatments with other chemicals that you can do. But you should first diagnose what you got. You need to know what you're treating before you start treating it. So you kind of got to be a little bit of a sleuth and a little bit of a fish vet. And unfortunately, that's not this session, so that's another time. We also cover our tanks, but we do have lights off in the corner, so we provide dim 24-hour lighting, but we keep them covered, and that's to reduce the stress on the fish. Because just like you guys, I mean, if we pack you all into a room and we had bright lights and things going on, and you get stressed out and you wouldn't want to be here. So we want to provide a good environment for the fish. I think that's the important thing to remember is, treat them with respect and give them a good place to live, give them good food, and they'll reward you with good growth. Then you can eat them. And we monitor daily. And we also monitor all the time. We're on call 24 hours a day, 365 days a year. And got to take this seriously if you're going to be successful. So one of the questions we always get is, when should I feed? And what is button up stage? So when we talk to people and we say, well, you feed your fish when they button up. And they look at you with this dumb look on their face and you're like, okay, button up means this. So if we look at these fish, the one on the bottom is a fairly newly, that fish has been in the heat trays a little bit. It's absorbed some of its yolk sac, but it hasn't absorbed all of its yolk sac. The one above it is closer. You can notice it's more developed as a fish. The chromatophores or color cells are starting to encase the yolk sac. And then at the top, it's fully encased and it doesn't have much of a yolk sac. So I would actually begin feeding at that middle stage. We would start to introduce fish feed to those fish. And we don't just start dumping feed in, we introduce it to them gently. We put it into the water, we submerge it, we squeeze it in our hands, we kind of let that smell get to them, and you'll see them get excited. They'll get excited and they'll be like, oh, where's that food? Where's that smell? It's like when you walk by a bakery, and that's what they do. And so you introduce them to the food gently, and then you start to increase the food. So by the time you're at that top stage and they're buttoned up, they know what food is, and they know what that is. If you wait till that stage, it's too late a lot of times. And believe it's funny, I've had some of the biggest trout hatcheries call me and say, I'm losing all my fish, what happened? Well, they waited too long to feed them, and it's too late. And every species is different. These Atlantic salmon we got, they don't want to come up. They sit on the bottom of the tank, so nobody wants to feed them because they're thinking, well, they're not coming up, because a lot of times they'll come up looking for food. So they're not coming up, so they didn't feed them. They die. What happened? You still got to feed them. So you got to be aware of what's going on with your fish, and you got to understand the species. Emma says this, and you got to know the species you're raising, the biology of the fish that you're raising, and what it wants. So that is the end of my show.
Work at the West White State Fish Hatchery. It's a Wisconsin DNR run facility. It's a cold water facility, so all we do is trout and salmon. Our main focus is lake trout and splake. Those are eggs we collect ourselves out in the Apostle Islands, and that's going to be pretty much the whole thing of my presentation will be the lake trout egg take. But we also do Cephorellin strain brown trout, about 175,000 a year. And then sometimes we'll do coho salmon, Chinook salmon. It all depends on what other areas of the state need. We all work together as a unit. We have 14 facilities throughout the state. Some facilities maintain broodstock. Our facility does not. Our broodstock is essentially the wild population out in the lakes. So yeah, our facility is old. Raising fish dates back to the 1897. That's when the brownstone building was first put into production. But actually raising fish on that property goes back to 1860s when Captain R.D. Pike owned the property. He saw the impact people were having on the fisheries, and he had commercial fishermen way back then bring them lake trout eggs, whitefish eggs, and he would hatch them out and eat stock mass sac fry. Survival wasn't the greatest at sac fry stage, but when he's dumping 21 million fry, that's a good number of fish. So So our lake trout program, that's a fish restoration project. We've been doing it for all of 18 years that I've been with the state. And this specific project is reintroducing lake trout over into western Lake Superior, outside the city of Superior. So the biologists come up with the numbers for us. We want 90,000 yearling lake trout stocked into western Lake Superior. Yearling for us is about 18 months old. So 18 months down the road, we have to make sure we have that 90,000. Three things that we take into consideration is the hatching rate. Generally, it's about 85%. Sometimes it could be 90%. In the sac fry, once they hatch out the sac fry stage, you'll see deformed fry, weak fry, Dropout for us is generally about 7 to 10% for trout. Salmon, it's more like 3 to 5%. And then finally, growing up in a facility, like you all know, they're a live animal, there's going to be mortality. As long as you have an accurate inventory, which begins in the egg room, you can calculate the right amount of feed and the right amount of space to give the fish. For us, in the 18 months, we'll lose about 5% of the population. And about 80% of that number will be, they'll die within the first 3 months of growth. So by the time they're 18 months old, 100,000 fish in a month's time, we'll lose 19 fish. So, but anyway, we go out into the Apostle Islands for our lake trout eggs. Our main target is Gull Island Shoal. Gull Island Shoal is just a little bit beyond Gull Island, but then also Gull Island and the northeast corner of Michigan Island. There's also other known spawning reefs, like off Cat Island, and I believe Devil's Island, which is up, well, yeah, it's up to the northwest. But anyway, Gull Island Shoal, it's a 19-mile boat ride. The boat leaves dock at 5 in the morning, 5:30. It takes about an hour and a half to get out there. This is the boat we use. We have a research crew that their main purpose is just to do netting surveys on the lake. Right now they're actually doing gill surveys going towards the city of Superior right now. We're hoping that next week they'll be in Superior by Tuesday so we could start stocking our yearling fish on Wednesday. With the weird spring we've had, everything is behind schedule by about 2 weeks. Ideally, all the yearling fish should have been stocked out by now. But it's a 55-foot, 1950s commercial fishing boat, 16-foot wide. As far as the commercial boats go, this is a nice one. Everything's enclosed, has a furnace, has a microwave. The pilot house has nice seating for the entire crew.
How long does that ride take?
It's about an hour and a half, 2 hours. Generally time it to get out there right as the sun's coming up. So you get some pretty sunrises out there. And hopefully it'd be nice and calm out there, which it usually is not. And yeah, for us now, the busy time of the year begins in October. That's when the lake trout are spawning. Generally, we shoot for October 14th, October 15th. Unfortunately, last fall was also delayed. We actually had to go into November to collect our eggs. One of our techs has been there for 25 years, and that was the first time we ever had to do that. But I have a short video, about 7 minutes. Is there speakers?
I think it would just be whatever.
Just on the, okay. Okay. Oh, where do I? Oh, okay. Oh, there we go. Oh, shoot. Okay.
There we go.
Okay.
Turn up the speakers. Is this speaker on this?
Okay.
There we go.
All right. I probably can't hear it too well, but I'll just kind of explain as they go through. The gill nets they set on Gull Island Shoal are 2,700 feet long, about a half mile. And as they lift up the gill nets, they're bringing in the right males and females. There's usually 2 to 3 guys picking off the table. There's a real art to picking the fish out of the net. I can't do it, so I just stand back. The gentleman in the background, he's actually our hatchery supervisor, Darren, and that's where we usually stand. But they're bringing the fish in. He's looking for the ripeness of the fish, whether it's male or female. He's looking for lamprey scars. And he's also looking for fin clips and also tags. Because, let's see. Okay, and that tool he's reaching, it's just like what you use to tag clothing in a store. Inject the Floyd tags to them. Yeah. And yeah, what they'll do is that was a recap fish that was already handled previously, so they just throw it back out. But as he gets right males and females that haven't been handled before, he'll hand them off to the to us. And of course,
if anybody wants to see the tagging tool, in fact, we can tag people at that.
oh sure, yeah, there you go. So yeah, and you'll see some nice sized lake trout. Anytime we get a lake trout over 42 inches, we'll hold on to it and take pictures with it before we release it. But we'll generally, on a good day, we'll handle maybe 350 fish total. Unfortunately, not all of them are going to be ripe. Yeah. Okay. But it's a noisy, I unfortunately can't hear the speakers, but there's a constant drone to the ship. And then you're below deck, so you can't really see the sunrise or the horizon. And yeah, so the way we do the eggs is we do a double disinfection. We actually use iodine in the water hardening stage. We'll use 75 parts per million. And yeah, we actually have a bowl with a colander sitting inside because we want the ovarian fluid to drip through. We'll actually take samples of the ovarian fluid of every fish we use, and it gets tested by our state vet. So if ever there's a disease found or what, we'll just throw the eggs out. And that's a nice female. A lot of times, the biologist will hand a fish back to us calling it ripe because he got one egg out of it. Over time you get a feel for when it's ripe, when it's not. And it'll be a rock hard abdomen. And if you squeeze too hard, the eggs just kind of pucker. We call them grape clusters at the vent. And we don't even take eggs from those fish. You like them to just flow freely. So for a quota of 90,000 yearling lake trout, we have to collect about 124,000 eggs, about 38% more than the quota. So, okay, so I think okay. Yeah, so, and that's a little variation, like he put the milt in and then added water and then stirred. What I'll usually do is add the milt in, stir it in, and then add water. We also disinfect it before we bring it into our facility, 100 parts per million for 10 minutes. And then this is, we still do it this way just to compare it to historical data, but it's a Von Baer trough, 6-inch piece of aluminum. Count the number of eggs. For lake trout, it's about 27, 28 eggs. And extrapolate the data into the number of eggs per quart, which is about 6,600 eggs per quart. And what they used to do is by volume, put a little bit of water in a volumetric flask, and then fill it up to 1 quart. The way we do it now is by weight. We do sample counts. This spatula has 100 holes, so we count over 1,000 eggs at a time. And we'll do maybe 2 counts to take an average. And it's pretty similar to, going to the hardware store, buying nuts and bolts. You can do it by the pounds. Same basic concept. And we go back and forth between metric and standard. I like the gram scale with the eggs in the fry, but the bigger fish we use just a regular scale. And then we just have a digital bent scale. We zero out the tray. We use Heath trays. That's all we ever used since I've been there. We just gently dump the eggs on. For lake trout, or for trout in general, it's 12,000 eggs per tray. For salmon eggs, it's more like 10,000. Yeah, and here, 4.66 pounds. It was such a small lot of eggs that we just weighed them all at once. But generally, it's about 3.2 pounds per tray. It's a very effective, efficient way of putting the eggs up, because once you bring them in, you want to work them up quickly, get them in the incubator. At our facility, we use 4 gallons per minute of well water coming in to each of the incubators. We do use the chicken waters for our hydrogen peroxide treatment. Yeah, once we put them in, it takes about 30 days for the eggs to die up. And then it takes a total of about 63 days for the eggs to hatch out. Yeah, from the time we put them in the incubator to the time they're starting to hatch, we're giving them a treatment of hydrogen peroxide each day. The chicken water has a drilled hole in it for a 15-minute dosage of 500 parts per million. So we put 325 milliliters of peroxide, and it's hydrogen, it's a food-grade hydrogen peroxide, 35%, which, yeah, you want to keep under 40 gallons on hand. Otherwise, you have to, yeah, we got it into a little pickle years ago. We had like 350 gallons or 400 gallons of peroxide. Yeah, Department of Homeland Security doesn't like that. So, and it was unfortunate because, you know, the peroxide you can only use, as far as I understand, you can only use it from Western Chemical. They went through the NIAID to get it tested and okayed. And it was only in 55-gallon drums, which doesn't make sense in a practical use. So fortunately, they finally got the 5-gallon carboys that they're producing. So we'll get 8 of them at a time, keep 40 on hand. Yeah. So last fall, like I said, it was kind of a weird fall. We ended up collecting 175,000 eggs, which is more than what we were hoping to get. Because, you know, each time we come back, we have a running tally of the number of eggs we have. But when you're out on the boat and things are coming on fast, you just, well, maybe I should take one more just to be safe. And it turned out to be a good thing because we actually lost a few more fish than what we do on average. So things evened out. But that was out of 42 females, 48 pounds total. The number of eggs per female came out to just under 4,200 eggs, which is a little skewed. That's, you know, the range was 3,200 to 6,000. 6,000 is an awful lot of eggs for the females. It's more closer to 3,600 for us, like the number of eggs per pound. Yeah. And then finally, we always get a picture opportunity with the big fish. So I guess that's all I have. Does anybody have questions? Yeah.
With the gill netting, do you have high mortality rates with the fish?
You know, we do have mortality, but it's not bad. It's like 5 to 10%.
Can you still, even if they're dead when they come up, you can't milk them at all? Or?
You know, we have to try.
Yeah.
A few years ago, where because sometimes if you hit it good, you'll get your eggs in 2 days. Generally, it takes 3 to 4 days. Some years it takes 2 weeks, and it's all weather dependent too, because a lot of times, you know, the storms in the fall are pretty bad and we can't get out there. But then sometimes, like this year, when we were getting there, the females were running late. You know, you handle a lot of green ones to begin with, and it wasn't until the end of October that we got the ripe ones. But yeah, so there was one year in particular that we actually took eggs from like 4 or 5 of the fresher females. And that's frustrating because sometimes when you go out on the boat, especially if you're the first day, it might just be a boat ride because all you get is one ripe female, and it's not worth bringing back 3,000 eggs.
You're at the northernmost research facility that the university has. And so we work with lots of different systems, lots of different types of fish. We're going to take a look at these guys. Actually, that tank right here, people can take turns going up here. You can look in the window. These are Atlantic salmon. These are Cascade strain Atlantic salmon. This is one of the few places in the world that we have them. The only other place is one of our partners. And we've been working with these fish for about 6 years. We pretty much brought them into Wisconsin to get this species going because we feel that it's a high valued species, a high dollar species, and it's a future for aquaculture. Any salmon you get when you go to a restaurant, most of the time it's going to be an Atlantic salmon. Right now a lot of them are raised down in Chile in research systems, and we want to promote Atlantic salmon culture in the US. So we're working with folks like the Conservation Fund, Atlantic Salmon Federation, folks that are interested in moving Atlantic salmon stuff forward. So these fish have been spawned several times already. As you saw in my talk, they're getting quite large, they're getting quite difficult to spawn. The number of eggs we get though out of these big females, we're getting 20,000 eggs out of a female. So if you can remember what Mike talked about with those lake trout, he was lucky to get 4,000 or something. We're getting 20,000 out of these fish. And they're high quality eggs. The company we work with is called Reproduction. They're in Washington. It's a private company, and they are going to be challenging or competing against Trout Lodge to supply eggs. They have coho salmon, rainbow trout. They're going to have Atlantic salmon now from this broodstock. And they're going to be selling Atlantic salmon eggs this year, 2018. I believe this fall they're going to start. But they have rainbow trout eggs and coho salmon eggs already that they've been selling. And their genetic program is pretty robust. So it's kind of interesting between Trout Lodge and them what's going on.
What was their name?
Reverence. They're in Washington. Right. So these are also the fish that are being used for some Atlantic salmon work here in Wisconsin, like at Superior Fresh. They're utilizing some of the Cascade strain, and supposedly these fish will get up to 10 pounds in 2 years. So that's kind of what we're waiting to see. But so far, they seem to be meeting those that growth curve. So this system is a partial research system. So you notice we've got a couple recirculating lines coming in, and we've got a freshwater line coming in. I guess from facility standpoint too, I like Hayward valves. I like true union valves. This is the kind of stuff, I mean, so if this valve goes bad, I can pop in a new valve body. It's not a big deal. I don't have to shut everything down. I can do that quickly. If you buy cheap valves, so a lot of people are like, man, those are expensive. You know, that's a $60 valve. But that valve has been there for 5 years or something, and they still work. If I open them and close them, they still work. That one's leaking. But I think that's something else, not the valve. And the nice thing about them is that they just last a long time, and I can trust them. I don't have to worry about them. You put a cheap valve in there, and I don't know how many places I go and I see that these get broke off on the cheap valves. They stick, they don't work, you can't move them, you force them, you break them, now you got a problem. And I see that everywhere. And so that's just a pet peeve of mine. So we use Hayward True Union valves. So this system comes over to here. We've got a drum filter, Hydrotech microscreen drum filter. So it's screening out the solids right here. The solids get sent to our settling pond, and the cleaner water goes through and is pumped into a biofilter. We're monitoring this with temperature and oxygen monitors, and those are all tied into our SCADA system, our monitoring system, and that is what goes to our alarm system. So when we get an alarm call at night, it could be the oxygen is low, or temperature is wrong, or the pump quit working, or it could be any of those things. So this water gets pumped over here. I don't know where that came from. So this is Caldness media is what this is called. This is a plastic media that serves as a biofilter for that system. So we have bacteria that lives on this, and that bacteria breaks down the fish pee basically, and then puts it back to where the fish can use it again. So the water comes through here. It's UV treated right here. We also add oxygen right here and an LHO, so it's a low head oxygenation unit. And then from there it comes around the back. We've got 3 pumps that are driving this water back to the tanks. And then we've got a flow meter as well. And all this is tied back into an alarm system and back to our SCADA system. And we also have air going into this system as well. So this unit was kind of meant to be a self-contained plug and play type unit. So the company that sells these, this is just their way of doing a biofilter and a RAS system. And I'm not going to say if it's the best or the worst way, it's just their way. We have done some studies on this and we have our opinions, and I'll talk about that maybe in a minute. But it's a pretty simple system, pretty easy, and because of the partial recirc, we're able to use a lot less water than what we normally would. We're only running about, when this was at full capacity, we were running like 60 gallons a minute to all the fish, and all the whole system was loaded with Atlantic salmon. And if we didn't have partial recirc, we'd be running about 400 gallons a minute. So that was a big savings of water. So we're stretching our water as far as we can. So Kendall's going to talk about this, but I just wanted to show it to you. This is one of our larval rearing systems. So there's like 29 tanks in here or something, and we can do replicated studies. And he's going to talk more about it, but I just wanted you to see it, and he'll fill you in on what everything means. But they're all custom made, replicated tanks, and this is where we do a lot of our diet studies and more research oriented stuff. This is another recirc system. This is our cold water recirculation system. We've got these 6 8-foot tanks on here. These are all Cornell style double drain tanks. And so what that means is that there's a bottom drain and there's a side drain. And if you peek in here, you'll see Atlantic salmon. They're in that tank too. And so these are Atlantic salmon. We've got a small project going on. We're looking at some water chemistry parameters with these fish. Biofilter, that's a sand biofilter. So that's filled with a specific size sand, and it's like a tornado in there, and the bacteria lives on the sand. And for cold water, the sand biofilter is the most efficient that we found compared to the plastic versus the sand. The sand is more efficient in cold water. So if you're looking at trout and salmon, cold water's probably better. If you look at other species, you might be able to utilize the coldness depending on what you're looking at. The water from there, from the biofilter, goes over to the CO2 stripper and then the LHO, and then it's fed back overhead to the tanks. So everything's above ground in this facility because we brought this stuff in after the facility was built. Not always the best thing to do, but we didn't know ahead of time exactly what we were going to do. These sumps are above ground sumps. I would prefer to see these in the ground, and then we could limit some of the pumping. But it's working very well. These systems have been here for 15 years running. So we do different projects in them. We have replicated. The tanks are replicated, so we can do 3 different setups with like densities or feeds or whatever we need to do, depending on how many replicates you need, I guess. If we look over here at this system, you guys can take a peek in here. Go slow.
Hey guys.
Hello. So these are walleyes. Well, yeah, they don't like that.
These are last year?
These are almost a year old. Almost a year old. These are not even 12 months yet. Yeah, so we have fish that are actually over a pound in here. These are part of the study that we're doing with Chris down at the AIC, looking at the different setups, the biofilter setups and the aquaponics setup. So he has the same fish in his setup with aquaponics. We have the same fish with the sand biofilter in our system. And we're looking at different densities and some of the water quality parameters. And so it's been really interesting. There's a lot of intensive water quality work going on. And you can see the fish are doing really well. These fish have all their fins too, which is a big plus for us. Some of the pictures you saw, the fins didn't look so well. So we've been fighting that a little bit, but these guys, they even have the white tips on the tails. So these have been raised totally inside, but we're figuring out some of the answers to those questions.
So how you get them so they don't eat their tails or lose their tails?
That's secret information right now.
All mine I got from you guys they all have missing at least half their tail now.
Yep. We've had issues. So some of it's related to flow, how fast you're spinning it, and then also how it's spinning, and then density. So we're figuring that out. But we've got fish at low density, which is 30 kilograms per cubic meter. That's what these are. We've got medium density, which is 60, and then we got high density, which is 80 or 90. I think it's 90, yeah. So we're feeding them with these 24-hour feeders. The other thing you notice, we got the tanks open now. We used to keep them all in the dark, and then every time the guys would come to work on them, they'd just freak out. And we were like, well, what's causing that? It was us. So we don't normally have all the lights on, but we started just leaving these open, and now you can walk up and they're all sitting there and they don't freak out so much. So it's just learning what the fish want. And that's where I think we're getting better at raising these, and so that might be why we're seeing better growth out of some of them. It might not just be the saw guys versus the walleyes, it might be that we're getting better. So, but we've been at it for a few years, I guess now. I think I've been raising walleyes for 30 years almost, so, but not this way. Finally got it figured out maybe. I don't know, so we got belt feeder, and we're feeding 24 hours a day. We got in-tank lighting to give them a little bit of lighting. And that's a license plate bulb. It's a 12-volt bulb, so that if there was an issue, it wouldn't electrocute the fish. And fish do get electrocuted. So this is a sand biofilter, just like the cold water setup. It's very efficient. And it goes over to a CO2 stripper. We have an oxygen cone on this one because it's warmer water. So we're running about 23 to 24 degrees centigrade here. Over there, we're running about up to 15 degrees centigrade. That's probably our max for those guys. We get a lot better growth even out of the cold water at that warmer temperature. These are baby Atlantics in here from our own broodstock. So these are Cascade strain Atlantics. And we kept some just for fun, and it's weird, so they stay on the bottom. They don't want to come up. And it's interesting, you know, we've looked at gases, we've looked at different things. We had them in an insert tank, but it's just the way they are. They don't like to come up a lot, but they will eventually. But the problem is you got to recognize that they're not coming up for the feed, so in the beginning you got to feed to them. You got to get the feed to them. And they aren't like rainbows or cohos, or they're weird.
Usually seeking.
So we're using BioVita, I think, right now. And in the beginning, we put them in an insert tank, and then we make sure that we're getting it to them. And then once they get big enough to where we can monkey with them a little more, then we move them into these tanks. And it's a sinking feed. We don't use floating feed at all here.
That's what I remember you saying, like you kind of put it in the water itself as well.
Yep, kind of squeeze it. For our systems, the sinking feed works the best because in those tanks, it gets removed very quickly if they don't eat it, so it's not sitting in the tanks. So, and the last thing are the heat trays for me. So heat trays, you lay the eggs in here, water comes in, bubbles underneath, comes over the top, goes down to the next tray. And we don't have any eggs to put in here right now, but you can get the idea. And depending on the species of fish, you can have different numbers of eggs, but probably not more. I don't usually put more than 10,000 at a max, depending on the species, and usually about 5,000, just so they have good oxygenation. We run about 5 gallons a minute into ours.
Walleye eggs come in. You can see these are eggs. They're in the process of hatching right now. Our setup, because we work with different strains, different species at times, we're capable of hatching 3 different groups of fish here. So they're compartmented out. You can see every tank. So every one of these tanks has a different strain of fish in it. These are the Mississippi River walleye. These are the Iowa walleye. And these will be sawguys. And we compartmentalize them out. So when the fish come in, we generally do a displacement method to volumetrically identify how many eggs we have. So we'll put the eggs into a known volume of a graduated cylinder. How much water does that displace? Pour them out, fill the jars. In this case, you can see this was 2.5 liters of walleye eggs that came into this jar. We'll then collect a subsample of those eggs. We can do the same process at a smaller level. I'll usually use a milliliter, displace out a milliliter, count the eggs, 170 eggs per milliliter. So we know that every liter has 170,000 eggs in it. We'll then look at that under the microscope, take a picture if need be, and identify fertilization success. So you can look at them right from 24 hours post-fertilization. Once they've all water hardened up, you can start seeing the cell caps developing, the fish is growing, and you can count and identify a fertilization success. So we'll put 2.5 liters in here. If we do the math quick, 340 and another 80 or so, so 420,000 eggs in there. 80% fertilization success, so we should have a little under 400,000 fish fertilized eggs ready to hatch out of there. We put them in. You can see that the jars are fed from the top. There's a tube. The jar has a rounded bottom. The water comes down. And what we're looking for is just a nice gentle roll. So we want these eggs to slowly move from bottom up to the top, and then they'll sink back down and slowly roll around. And you can see after a time, the white eggs up here, these are all dead eggs. It's too late to kind of work them out now because everybody's hatching. But they'll, after a few days, they'll segregate out. Dead eggs will float on top. You can take a tube and you can siphon off all the dead eggs. You want to get them out. We're concerned about fungus growing in here, and of course attacking the good eggs. The dead eggs is a perfect habitat for the fungal spores to grow on. We get them out. Nice gentle roll. We start our temperature at about 10 degrees C, and then after a period of time we'll slowly start increasing that. They're at 12 today to help accelerate the hatching. You can hold them at a temperature for a bit of time, delay hatching if you have something you need to wait for, or accelerate hatching mostly by water temperature. But you can't overdo it also. You can wait too long, they want to grow, they want to hatch, and you can try and go too fast and they don't have enough time to fully develop before hatching. So there's a window in there of anywhere 7, 10, 14 days based on water temperature.
How long will it take for that one jar of hatching to fully hatch?
Now that we're increasing the temperature, it won't surprise me, this will be empty tomorrow. They started hatching about 2 days ago, 1 or 2 going on. We increased the temperature from 10 to 11, accelerated it a little bit. Today we went up to, I got 11.8 in there, and it won't surprise me if it's mostly empty tomorrow. You can see we lift the screens. That allows the fry to swim out. You'll have some in here. You're going to have some that aren't going to hatch. You're going to have some that aren't strong enough to swim out. But you don't want those fish anyway. okay? Back to even, this is the idea behind the lights. So when it comes time to stocking, we can slow this air down a bit. This is just helping keep the screens clean. Eggshells and stuff aren't building up and then overflowing our tank. We'll end up siphoning this clean as things progress.
So you siphon clean it?
Yep, we'll siphon right underneath that. Generally right underneath there will be a handful of dead fry that get sucked in there. Some of the old eggshells as they start coming out. And then you just want to be able to keep this clean so that you can, a little brush on occasion. That way it doesn't plug up on you. And so as we slow this air down for stocking, then this light, the walleye are super photopositive at this stage in their life. So they're going to go to the light. And that's the fish we want to work with. Those are strong swimmers, ready to eat. They're up at the light. They're ready. They're looking for something to eat. Out in the wild, it would be rising up, looking for the zooplankton that are up consuming the phytoplankton in the ponds or in the lakes. And so those are the best fish. The ones that are laying on the ground over here or swimming in circles, you're not interested, why bother with them? So it's a selection of the strongest to put in our systems. I'll add also, we occasionally have to treat these eggs. So we got flow, you measure up a flow rate, how much water is coming into the system. Then this has the example on it. This is it's a general recommendation. We work with formalin, all right? It's a 15-minute treatment between 1,000 and 2,000 parts per million. It helps slow down fungal growth, okay? Doesn't hurt the eggs so much, but retards fungus. So this chicken water with a little hole in it, this is 15 minutes worth of liquid. So we fill this up, that's 15 minutes. Our basic recipes right here, if we're running 5 gallons per minute, that's if we're running 5 gallons per minute, and we wanted a 1,600 parts per million treatment, then we would put 473 milliliters of formalin in here. Then you fill it up, you cap it up, you tip it upside down over here, and that'll distribute that 473 milliliters over the next 15 minutes through the eggs and out. You can do that to a certain point. Once the eggs get very strongly eyed, they're ready to hatch, the formalin's toxic. So you can see we can't do much about anything at this point in time. We can't get in and siphon these clean without taking off too many live fry. We can't treat for fungus that may grow in there, some clumps that start. But right now, we're ready to get them here.
You know what size hole you have on the bottom of that?
No, I do not. I've measured it maybe one time.
I use a peristaltic pump when my pumps go out, and that's a really easy, quick, cheap.
Yep. And so, and mostly the way to design one of these is you just punch your small little hole. This might be like 3/16, okay? Yeah. You fill this up, you tip it upside down, you measure how much water comes out in 15 minutes. Now you know that volume. You measure the flow rate you got in here. I use an Excel spreadsheet from the American Fisheries Society, Fish Health, and you punch your numbers right into that, and it tells you how much formalin you need to add to match that flow rate for 15 minutes. But this is essentially just the timer. The peristaltic pump, same thing, right? You measure how much your pump is going to pump, how much do you need to add, and adjust it. Absolutely. So this is the setup. This is how it looks on our morning coming in here. Okay? This is our larval rearing. Dim lighting, water spraying around. Come in the morning, and on the early stages, you can come in and somebody can reach up top and slip that light on. There you go, so we can all see what we're doing. You can, early stages, you can turn the lights up before we start cleaning or anything. Again, the walleye are photopositive, and this will help get them up and off the bottom, as we'll find out why later in a minute. This is our arrangement. So you can see we're running a highly turbid water. This is a clay water. We have a head box up above you holding a couple 100 liters of water. You do the math how much that, how heavy that might be, okay? And then a clay sump here. So we have a recipe we've worked out. We put 4 pounds of clay for every 100 liters of water into this sump. We pump it with this pump at a 40% rate. And what I get out of it at the end, it pumps up into the head box. We use air. You can see I use air to keep the clay in suspension. We have air and if need be oxygen in the tank up above to help keep the clay in suspension. And we get this, we're shooting for 50 to 80 NTUs of turbidity. It's a special clay. It's a soft clay, so it doesn't really bother the fish much. It's straight out of Kentucky. And you can see, so we put in our clay water. The idea, the hypothesis we're working with is this creates this cloud effect. Black tanks, dim light, turbid water. We're trying to get these walleye to disperse throughout this tank. We can put upwards up to 10,000 fry in each tank. We're generally working with closer to 5,000 lately, but we've done as high as 10,000. It's a 60 gallon, a 220 liter tank. We have directional water flow. Just slowly right now, we're running 2 liters per minute, enough to get the water to at least flow. Hopefully aligns the fish, but not that they have to swim too hard against it. The clay and the dim lighting helps disperse these fish 3-dimensionally throughout the tank. Walleye are highly cannibalistic. They have a mouth gap that's capable of consuming something their own size, and so brother or sister next to them will fit in their mouth. Unfortunately, most of the time they can't digest something their own size, but they can try and will try to eat it. So the idea is we line them all up. We offer them our special food. This is the Otahimi. It's a really fine, it's a 200 micron size pellet, 24 hours a day. Cloudy water, we disperse them all over. They can't see each other. They're all lined up that hopefully we can reduce some of the cannibalism. Walleye must physically gulp air off the surface, break the surface of the water to inflate their air bladder. So these are just misters for hydroponics or even the grocery store vegetable shelves, disrupting the surface water, the surface of the water, the surface tension that knocks the oils that may come off of the feed and then gives a spot for the walleye to actually come up and gulp air. It's yeah, this is actually, I believe we have sizes on these. It's a silk screening cloth built around a plastic screen tube. They used to make better ones. And we have varying sizes. Should have also, the fish will be in here for 30 days at minimum. And this is our morning cleaning procedure. So again, we'll come in, check the turbidity, we'll check the temperatures, make sure everything's working the way we like it to. And we've developed this tool. There's other ways, it's just an excluder.
Okay, you gotta hold it up for me.
Oh, well, you'll come up with your own method of excluding fish.
Yeah, that's all good.
So we just slide this down, get this out of the way, maybe a little drop at the end. What we don't have available for you is an empty tank you could peek in. At the bottom of this is a 4-inch PVC pipe that's around the drain, and it's what's holding up these screens. So, yep, you take the screen off, pop in a new one, and then carefully remove this without pulling it up. So change all the screens, and then squeegee the sides of every tank. just to knock off any clay. You can see to the sides of the tanks. We just want to get the tanks cleaned up some. Fungus is still an issue in here. This is a beautiful environment for fungus, right? Everywhere. 115% humidity, nice warm water. So we'll go through, we'll squeegee out all the tanks. Then when we're done with that, we'll grab our fancy vacuum cleaner, okay? So this is still a work in progress. I'm sure there's some better ideas.
I'm liking the ideas. I need all the help I can get here. It kind of looks like ours.
Yeah, no, this was highly engineered tool. And like I said, most of the time, everyone gets their favorite because there's really only one way to do this. You start a siphon, and like I said, early on, so now we want these fish up off the bottom. We're gonna go through, you can see the dirt, the excess feed. You know whether you're down on the bottom. Sometimes you have to feel for it. Sometimes it works out just nice for you, and you come around the tanks. We usually drop these off, get them out of our way. Siphon them all clean. It takes a good 2 or 3 trips around here to get everything off. We're counting the fry. So now we'll take this siphon water, pour it through a soil sieve of appropriate size for the fish. Later on you can use a regular kitchen strainer. Pop it into a white pan, dish pan, and count. And so put on your cheater glasses or get a magnifier. We have in the other room, I have a nice magnifying lens that you can use. You see how small these fish are, and you're looking to find out how many of them have passed on over the course of the night. Upwards to 500-600 at a time, depending on the life stage.
What kind of paint do you use?
This is a tank paint.
Yeah, tank paint.
Yep, an epoxy tank paint for aquaculture. The other tanks, the new tanks we have, were ordered with the black clear coat. Okay. So, yeah, we keep wondering, this is the room I have set up and going, which is why we're using this. We have better tanks now. We have some tanks with a side standpipe. We get a bottom draw. We feel it get a better cleaning, especially at the later stages. Again, as these fish grow, we're swapping out screens. So here would be the next size. See, we got them all labeled. So, you know, the early stages, these fish are eating feed that's 200 to 300 microns in size. And then as the fish get larger, the feed sizes go up. We have a regular procedure we're trying to follow as far as increasing feed sizes for the fish, increasing feed amounts. Then we go to a number 2 screen. And then our next screen size is the plastic alone, without the silk screen wraps. So we're trying to get big enough to let the feed go through, so it doesn't dirty up the tanks too badly, but small enough to keep the fish inside.
I'll show you this computer screen first. Can you see that okay? So we're kind of a small facility, right? So this is kind of our lunch room and our break room, and this is our water quality. So we do a lot of our water quality samples here, but also our larval rearing. So we've got our microscopes in here. This is our SCADA system, this is our main screen for that. We also have an electrical room where we respond to our alarms, but this is the basic screen for this. So SCADA stands for Supervisory Control and Data Acquisition. So that's basically monitoring our farm. We have flow rates being monitored, water levels being monitored, our electricity and pumps. So a lot of fish species that you saw here are part of major projects. We can't afford to lose them. A lot of farms, you know, have alarm systems being set up now, especially when they're working with a lot of equipment, a lot of pumps. So we also want to have 2 of everything as well. So on site here, this is the overview of our whole facility. So we are on 40 acres, but 20 of that is developed. So we have kind of room for expansion if we ever decide to do that. But basically, this was Highway 13 here, so you guys drove in, you drove past our first well house. So we have 2 wells that fluctuate every day, so every day at noon they'll switch over. So we want to exercise, we want to make sure they're both able to run if we ever need to use the other one. So we can go into these and we're tracking how much water that we're pumping. So a major reason that we're here and that the Red Cliff Tribal Hatchery is here and Les Voigt and Iron River Federal Hatchery is because of this aquifer. It has really amazing water. It's very large and the water is quite pristine for raising fish. Fish just seem to do better here than at other places as well. So we have this great resource that we're using, but we also want to be careful and make sure that we're not overusing this resource. We were taking data on how much water we're using. We can share that. And we can compare that with Red Cliff and whatnot. And we also have a zone of influence too. So we know that this well is not affecting this well, basically, for the height of the aquifer.
Are they at different depths?
They're both, I think, about 360 feet deep. So they're pretty deep as well too for this area. These are also, so these are high capacity wells. They have the ability of pumping us with 800 gallons per minute, but we never reach that. Normally our highest is about 500, and that's when we have a lot of larval systems going that are flow through. A lot of our systems here are water recirc systems, so we're just reusing that same water. Most of this right now, that 300 gallons per minute, a lot of that is going out to our raceways, which you guys saw, so that's all flow through. A little bit of this trickles into our recirc systems, but that's kind of what we like to promote, is being able to reuse that water. These pumps also have variable frequency drives on them, so we never have to pump more than we need. We never have an overflow. So back in the day, you always had to pump more than you needed so that you never ran out, you never had an issue. We have VFDs so that if we open up a valve or start to use more water and our head tank level will drop, that'll trigger our pump to pump more water for us. So it's very efficient and effective. We never have to waste water. Inside our aqua barn, just briefly, did you see the flow meters on any of the systems? Did Greg highlight those? So we have paddle wheel flow meters, so that's what this is calculating. So in this walleye system, it's showing about 217 gallons per minute moving in that system. So if this number drops, meaning if a pump goes out, if we lose water somewhere, somebody left a valve open, we would get an alarm for that. So we have different alarms throughout the facility based on kind of what we're looking at. So flow meters, water levels. We have a hospital grade generator on site as well too, so if we lose power, that'll turn on pretty instantaneously, but we still get an alarm. We still have to physically come over here and acknowledge and make sure everything's okay. Do you guys have any more questions on the SCADA system or our monitoring?
Is the SCADA system, is it custom built just for you guys, or is that like something that a lot of places are accessible to? Like?
So this was built just for us, but a lot of the companies doing this used to be for water treatment facilities. So they still, were using alarms. It was just for a different reason. So a lot of them are switching over to kind of these aquaculture facilities, because more aquaculture is using pumps now, is using top technology that needs to kind of monitor these things. So LW Allen is the company that we go through, and they're out of Madison, I believe. So we can call them up and say, oh, can you change the levels on this, or change this alarm, or how it works for us, or if we have an issue that we can't figure out, they pretty quickly respond. So it's good to have kind of a working relationship with them to be able to, because a lot of the SCADA technology we don't know, we just know what's happening on our site. Yeah, when we went to Idaho for that conference, we said, oh, do you have any alarms for anything? And they said, we don't need to. The water pours out of the mountain, goes through our systems, it's all gravity fed, and we don't have to worry about it. Unless the water stopped coming out of the mountain, we don't have a lot of alarms. They could maybe hook into oxygen and temperature. And we do have oxygen and temperature probes out there that we could link to this, so eventually maybe we'll be able to see that as well. But you can get pretty infinite with the amount of alarm systems you can hook up with this too. I mean, you can be pretty, it can get a lot pretty quickly. But so I'm going to move over to this program here. This is called CellSense. So a lot of the photos I had in my presentations and videos came from this program here. So what this is, this computer is hooked up to a camera and a video on our microscope here. So we can share a lot of data when it comes to larval fish. We can take a lot of measurements that we weren't usually weren't able to do before. So before, you're looking at these larval fish under a microscope with a ruler trying to figure out differences in sizes or egg sizes, and it's nearly impossible. With this screen, we can calibrate it, so we can take measurements right on the screen, and we can save things like videos, photos, and things like that. So this photo is showing kind of egg fertilization success. So these are 24-hour post fertilized walleye eggs. So we can see that all these are have been fertilized, and what we're looking at is egg mitosis and meiosis. So this is cell division happening on one side of that egg. So we can see most of these are fertilized, and then we do have a few here and there that are unfertilized. But this is generally a good fertilization success. And what that looks like, you guys saw the bell jar systems, but you can see which ones are fertilized and which ones aren't there. So again, the ones that are dead are white. They generally float up, and that's when we can siphon them off the bell jar system. And Greg mentioned when these fish become hard-eyed or very darkly pigmented, they're ready to hatch. So we normally bump the temperature up a couple degrees to get them to all hatch at once at this point. So you're seeing that happening right now. So they become really active. You can see some of these will start to move around inside the egg. And the egg becomes pretty malleable and kind of almost disintegrates off, and they start to move around and try to hatch out of that. So this is a different species. You guys want to take a guess at what those are? Perch, right. So how'd you know they were perch?
They're in a matrix.
So if you guys notice, they're all connected, right? So they're in that gelatinous matrix or ribbon. So they're all connected. So to get these guys to hatch all at once, we use that paint mixer basically inside the bucket to get them to break apart. In nature, this would happen with waves and water movement and things like that. But we really want to get those fish out at once to be able to stock them in the system. And you can imagine how difficult it is to be able to count these fry, so we can't really put them through a larval counter because all of this matrix comes with them. It's really hard to break them away from that.
Will they die if they're stuck in the matrix?
Eventually, yeah. If they're stuck right in the middle. So this is just a cool video. It's just showing the heart and how the heart is beating and that larval fry. And still they look pretty underdeveloped at this point. Their mouth is just starting to form, but they're still kind of absorbing that yolk sac. And you remember this video? What is this? You guys remember? Sue, you know. So this is in the pond culture workshop, right? Or session, or overview, I guess. So this is our zooplankton levels. So this is the program that we use to check zooplankton levels in our pond. So basically using that plankton net, and then we bring it just a couple drops of that underneath the scope, and we can see how much of life actually happening in that pond. So we're fertilizing our ponds to get this plankton bloom, and that's what our walleye fry will be feeding on. So that's when we raise them extensively out in our ponds. They're feeding on that zooplankton. If we raise them intensively in our system, we start them on a commercialized feed, they never see live feed, never see zooplankton in our indoor systems. So this is a really cool video on milt activation. So this is using our high-powered microscope. So our Atlantic salmon broodstock, we wanted to make sure that the males were producing viable milt because we wanted to use a specific male with a specific female because they're doing this broodstock program. So we wanted to know the genetics of the fertilized eggs. So how do we find out milt activation? So milt becomes active when we put water with it, right? So what this is just a very small drop of milt on this microscope. And what we're going to be doing here is adding water just to the edge of that. And you can see how many millions of sperm cells there are in just that tiny drop of Atlantic salmon milt. So if this wasn't viable, if this sperm was essentially dead, it wouldn't be moving at all. But you can see that the minute that comes in contact with water, that sperm is viable. So this was a good fish to use, so we used that to fertilize our eggs. But you can imagine when you use extender for milt, I mean, one drop of milt will fertilize millions of eggs. This is just showing you kind of the difference in sizes. So, did I show you guys these yet? I'm getting confused with groups. So this is the size we're working with, right, with our walleye and our perch, kind of the size of your eyelash. And then these larger fry here are more of our cold water fish. So that's like our Atlantic salmon, rainbow trout, Arctic char. So much different techniques in raising them and incubating them. So our walleye fry, we have to get food to those fish in 3 to 5 days. With our cold water fish, they are absorbing that yolk sac for about a month to a month and a half. So we don't want to feed that fish until that button-up stage where they absorb that yolk sac. So this is a cold water fish. If we feed the cold water fish too early, they like to kind of sit in the bottom of that tank, and that feed can get into their gills and become irritated if you feed them too early, if they're not accepting feed. And then we don't want to feed them too late because they're going to start to starve. So we kind of find that kind of sweet spot right here where we start introducing feed, making sure they're accepting and all that, but much different than our walleye rearing. And then just the last picture I have here, this is a walleye and this is a perch. They're the same age, they've been raised the same way in our intensive systems. So this is just showing you why we're moving more towards our walleye in our intensive systems, because of that growth and that kind of that vigor, that hybrid vigor for the sockeye as well too. So the perch, a lot of people start perch out in ponds to get them going and then feed train them. In our intensive systems, there's still some key things missing. There's still this mortality burst that we're seeing. So we're trying to push more towards the walleye or the sockeye.
Is that the way market's going down also? Going more towards the walleye or sockeye?
Market for food fish?
Yeah.
Yeah, I mean, I think there's always farmers that just still want to try perch. They still want to start raising perch. But I mean, I think people still love to eat perch. They probably still would buy it, but it's just this issue in this larval mortality. Do you see markets? It's just...
Right now, there still isn't any farm in Wisconsin raising walleye as a food fish. Those that are raising them are raising them as stocking the fish. So if you do see walleye on the menu, it's probably wild harvested, most likely by Canada and the Great Lakes. You know, yellow perch, I think forever yellow perch, there was a study done 20 years ago or something that put a pin like in the center of Lake Michigan and then drew like a 60-mile radius around it, and it obviously hits the east side of Wisconsin. The people there don't want to eat anything but perch. You know, so there's always going to be that market for them, even if walleye become more available because they grow better. Most people still want their perch.
It's still the Friday night fish fry. People just still, it's a cultural thing almost.
As I say, based on this morning's talks, I think we've gotten the walleye production now down to a point where we think it can be profitable and productive for people. I think the next thing we've been talking about is that out-of-season spawning. It's got to take place so that they're available more than once a year. But that's still the same case for the most part for yellow perch as well. There really isn't a provider of perch throughout the year, maybe 2 times per year. So there's one more obstacle that's there, in which case we hope that somebody launches a business producing walleye, either as feed trained fingerlings or as food fish. Because we're not in the business to do that. We're in the business to figure out how to do it. So somebody else just has to do the investment and take on that challenge at that point. Hopefully that what we've done, we'll have we already have videos, we'll have more videos, and then we'll have a manual on how to do it.
And we're getting some economic data as well on this year, and kind of what does it cost to get there too, which is important for people, because it's really, nobody's done this yet. Nobody's done this commercially yet. I mean, they tried it for perch, but now there's this new species coming on for walleye. So hopefully within this next couple of years, we'll have that data for people.
As I said, perch still to this point is a pond-raised fish. If you do get farm-raised perch, most likely it came from an outdoor pond on a fish farm. There's some potential to do it indoors, but I don't know, based on the size, you'd be amazed at how much that smaller size gives you trouble from the whole process.
Right. So I have a short activity for you guys if you want to sit down. So there's a scrap piece of paper on there if everybody wants to grab one, and a pen. So we've got some BBs here that we're going to pretend that are fish eggs. And first I want you guys to take an estimated, educated guess on how many are in there, just from your smart brains. Abby, do you want to do it too? You want a piece of paper? All right, so if you got that in your head, write it down and flip your piece of paper over. So how would you figure that out? If you guys could do anything to figure this out, would you count every single one? What would you do?
Weight of so many.
Yeah, you would figure out maybe how many to a gram and then weigh the total, right? Anybody else? Any other ideas? Corey, you probably know how to do this.
Volumetrically?
Volumetric displacement. So what does that mean? So what we're going to do, so everybody grab a 10 milliliter graduated cylinder there and your eyedropper. And so what we're going to do is figure out how many eggs displace 1 milliliter. So put or take a known volume, so what we've been using is 5 because it's right in the middle, so fill your graduated cylinder to 5 milliliters. And if you guys know how to read a graduated cylinder, you have to look at the bottom of that meniscus, right? It's like my kindergartens I work with.
People coming out of school that don't know.
No. It doesn't really matter, just as long as you know where you start. So we say go to 5 because it's right in the middle and it's easier to see. So once you get it to 5, you can grab a handful of BBs and start adding enough eggs or BBs to raise that to 1 milliliter. So if you added 5, raise it to 6. Doesn't have to be too exact. So you guys got some numbers? What'd you guys get?
I got 21.
Okay. What'd you get? Okay. Anybody else got any different? 21, 22? Yeah? Nice.
I go with 22.
Okay. So about 20 to 22. So when you're done, you can pour your BBs in there. So now what are we going to do? So we know how many BBs displace 1 milliliter. So we need to displace all the BBs, right? So I've got a large graduated cylinder here. And I put it to a known volume of, I think, about 300, right? Abby, do you want to pour the rest of those in there? We want to get them all back together. And Abby, do you want to pour them in there for me? Good job. So what are we at? What's the difference? So we started at 300, and now, can you read that, Scott?
155, or sorry, 175. Am I wrong? Yeah. 175.
So this is 300 to 400 is 100, 150, 55, 60, 65, 70, 160 we'll say. So what do we do with that number? So if all of them displace 160 milliliters, we multiply that by our 22, right? So if 22 displace 1 milliliter, so what is 22 times 160, we'll say? We've got a calculator here, it's easier. I thought you were testing yourself.
I really
So what are we at? 3,520? What did you guys guess?
3,500.
Oh, that's pretty darn close. That was close.
You come to the fair with me.
Yeah, right? The gumballs. Yeah. Oh, nice.
5 away. It was amazing.
That's funny. Some people say 10,000, some people say 500, some people are like, I don't know. So this is exactly how we figure out walleye egg counts, right? So we have to figure out how many walleye eggs displace 1 milliliter. And normally it's about 150 to 160 eggs that we have to count to displace 1 milliliter of water. So you think about those bell jars that you saw, they fit about 400,000 walleye eggs in 4 liters. So the size is much different. This is a little easier and faster, but you can imagine, you know, how many walleye eggs you can produce. If you guys go to those big hatcheries where there's rows and rows, probably what you guys have. Sure. I guess you'll see that at Lac du Flambeau. They have quite a few rows of bell jars, but you can raise millions of fry pretty easily. It's just getting them to those next couple stages is the issue. Oop, I'm sorry. But yeah, it's, so you guys understand that? Yeah. About?
Yeah.
So you guys can use that at your next fair with the gumballs or something.
Yeah.
Right? Yeah. So I did weigh these before, and I think they were supposed to be, I think there was 3,290 or something. So we were a little bit off, but you know, everybody's meniscus levels are different and weight is a little more accurate, but that's how we do it for walleye fry.
So you guys use the counter for the walleye for the fry?
So in here we have 2 raceways. Ciscoette lake trout over here, lean lake trout over here. Looks like the leans are hanging out in the back. You can see some tags on some of these fish, pink, green, blues, grays. These fish are being used by a doctoral candidate from Michigan State University. He's looking at the effects of lamprey parasitism on lake trout. What he's looking at is a lot of things. It's a pretty detailed study. He's looking at mostly fecundity the next year after parasitism. So we get our lamprey in, mostly from commercial fishermen here that'll bring them in. We'll put the lake trout in these little 4-footers for 4 days. Once they parasitize the lake trout, we'll leave them in there for 4 days. We'll take them out. They get blood samples, fat meter samples pre and post, and fat meter readings. They'll come in here, back in here, and then we'll take them out the next fall, spawn the fish that were parasitized, and then we can look at egg, number of eggs, and we'll spawn them, so we'll look at how the fry do. It's a pretty in-depth, it's a lot of work, and we're out here in middle of October through January, so it's cold, like really cold, and we don't really have no way to heat the building, so it can get pretty brutal. These 2 raceways, pretty simple. Water comes in the front. We back here, we got some stand pipes. We can just pull them. All the water then is going out to the settling basin. Grab a broom, sweep them down, clean them up. Pretty basic. I don't know. Any questions?